Good morning, everybody. Thank you for being here this morning, and I want to welcome you to the 2019 14th Annual Lewis L. Judd Young Investigators Symposium. For some of us who've been here for a while, it sort of seems like that reads the 201,914th Annual Lewis L. Judd Young Investigators Symposium. But this morning we have 8 speakers, followed by a poster session with over 100 posters. We're streaming live to an audience of millions, and so I want to welcome you all here this morning. Lewis L. Judd, who was the chair when many of us joined the department, the second chair of the department and former director of the National Institute of Mental Health, was a powerful, strong, compelling advocate of biomedical research and psychiatry. He established the department as a research powerhouse that it is, and really made as a top priority the development of young scientists and physician scientists, and we honor his legacy this morning through this annual symposium, and you'll be hearing some more about that. In addition to the presenters, we have many faculty mentors here in the department who will be joining in the teaching process during the poster session. I'll talk a little bit about that. Are there faculty mentors here who could stand and be recognized? Any of you here? It's early in the morning. Okay, here you are. Welcome. Thank you, all you faculty mentors. You're each assigned a group of posters, and the poster presenters will be meeting with them after the talk session this morning. In addition to the faculty mentors, I think you all know the people really pulling the strings behind this meeting this morning are the ones who've done all the hard work. Jennifer Hickey Hinojosa. I don't know if Jennifer's here, but if not, you'll be seeing her later this morning. And Sean Rybeck, who's running the show behind the monitors in the back there. And thanks to both of you. Sean, can you move us to the next slide? Thanks. So this is the schedule for this morning. There'll be some welcoming remarks followed by 4 talks and then a brief break and another 4 talks. There'll be time for questions after each talk, and then the last talk is the plenary, which is a little bit longer than the others. We'll leave a little bit more time for that. 11:30 we adjourn over to the poster session and lunch across the hall in the MET. And there'll be 2 periods of time that we're going to use for the mentoring process. So if you're in posters 1 through 52, that's the first half hour, and posters 53 to 106, the second half hour. As in all large productions, there are last-minute adjustments. We have some mentors who've been stuck in planes and airports overnight all around the country, some recovering from a schizophrenia conference that ended late last night. So there'll be some small changes to the program. The part of Vince Filateo will be played this morning by myself, Dr. Swerdlow, and the part of Jared Young most likely will be played by Dr. Light, so you can make those adjustments in your program. And so it's now my honor to introduce the next introducer, the interim chair and professor in our department, Dr. Robert Antonelli.
Thank you, Neil. Pleasure to be here with you today. I am Robert Antonelli, and I'm going to echo some of what Dr. Swerdlow mentioned to open our remarks today, and that, this 14th annual Lewis L. Judd Investigators Young Symposium is really special in at least 2 ways today. Since we lost Dr. Judd in December at the age of 88, this is the first symposium to honor him posthumously. For those of us who knew Lou and had the good fortune to work alongside him, I think we all know how proud he would be today of relishing in our achievements. We were part of his extended family. He knew that the life's blood of the department, that Arnold Mandel and he founded, was in passing along the baton of scientific knowledge from one generation to the next. He also knew that the decade of the brain, a term he coined when he was the director of the National Institute of Mental Health, would extend far beyond the 1990s and into the 21st century. The junior faculty, postdoctoral fellows, and trainees presenting their work today are part of Lou's legacy, and we're the beneficiaries of his vision to launch this annual research symposium 14 years ago. The second reason why today's symposium is special is that it coincides with the department's golden anniversary. I know many of you took part in the 50th anniversary symposium from April 4th to April 6th, and I hope you left that experience with the same one that I had, and that is, damn, that department is good. But beyond the display of top-flight science, stellar educational programs, and cutting-edge clinical practice, what shone through to me was how deep our department is in up-and-coming young scientists whose work we'll proudly display today. Finally, I'll close with 2 thank yous. I first want to thank Neil Swerdlow, Greg Light, Jennifer Hickey-Hinonosa, and Jyoti Mishra for their hard work putting together today's symposium. So thank you very much, the 4 of you, and also Sean Ryback for putting today's together. I'd also like to thank our outgoing chair, Dr. Igor Grant, who I think is here somewhere. Igor, are you here? There he is. Hello, Igor. For shepherding us through not only the transition from Dr. Judd's chairmanship to his own, but for also leading us and placing us on solid footing as we contemplate our next 50 years. Thank you, Igor, for your wisdom and outstanding service to our department. So with those comments, I will end and introduce our next speaker, Dr. Gregory Light.
Good morning, everyone. One of the traditions of the Judd Symposium is that the keynote speaker serves as one of the organizers and moderator for next year's session. Last year's keynote speaker, as mentioned, was Dr. Jyoti Mishra. Dr. Mishra is an assistant professor here in our Department of Psychiatry, and she will be running the show today. I'll also echo some of the comments from Neil and Rob that it starts tomorrow, organizing the next session, and that involves soliciting abstracts, collecting the abstracts, reviewing them, forming a committee, deciding on the order of presenters. So I really want to thank Jyoti for doing an excellent job arranging this year's session. And with that, we will introduce Dr. Mishra to kick us off.
Thank you all. It's an honor presenting all the speakers today, and thank you for the opportunity to help organize this. I felt like I was standing on the shoulders of giants, not really having to do too much, and it was really a pleasure looking at everyone's really impeccable work, and I'm really proud to be part of this department. So without much ado, our first speaker today is Dr. Himanshu Mishra. He's a stem cell scientist with long-term research interests in modeling neurodegenerative and psychiatric disorders using human-induced pluripotent stem cells, or iPSCs. He earned his master's degrees in India and also his PhD from Germany. And his PhD work was focused on modeling neurodevelopment and cortical dysfunction in motor neuron disease using iPSCs. Currently, he is a postdoctoral scientist in the lab of Dr. Michael McCarthy, and his research project involves studying the role of circadian rhythm-associated clock genes and the biological basis of lithium-induced neuroprotection in a human stem cell model of mood disorders. He has received the Best Center for Circadian Biology Trainee Award in 2018, and he was also an invited speaker for 2018 SFN Neuroscience meeting and this year's Center for Circadian Biology International Symposium. And today he'll be presenting on modeling neuronal circadian rhythms in bipolar disorder using human-induced pluripotent stem cells.
Good morning, everybody. Thank you, Dr. Mishra, for the kind introduction. I'd also like to acknowledge first the organizing directors for giving me the opportunity to present our work. Basically, our lab works on understanding the pathophysiology of bipolar disorder and uses human iPS stem cells to understand some of those cellular and molecular mechanisms in the disease. To start with, bipolar disorder is a common and serious neuropsychiatric disease which is characterized by episodic periods of mania and depression. It is associated with major changes in sleep, activity, and appetite. The age of onset of the disease is around 18 to 25 years, which also suggests there is a neurodevelopmental component for the disease. There is an important genetic risk for the disease, which in the first degree relatives is around 10%, whereas in monozygotic twins, it could be as high as 70 to 80%. But the main enigma of the disease is that the biological mechanisms causing the disease is still unknown. And that's why the treatment is based on trial and errors, which involves basically using the mood stabilizers, and the lithium is actually the gold standard so far in treating those mood in those patients. However, only 1/3 of the patients respond to the lithium, which are called the lithium responders, whereas more than 30 to 40% are either moderately or do not respond at all, which are characterized as lithium non-responders. So decades of research have already established that these patients have shown different characteristics of rhythms, disturbances, for example, the mood, which varies according to the sensitive to seasonal and daily changes in the light, the sleep, which according to the disease state, as in mania, could be decreased need of sleep, or in depression could be hypersomnia. Similarly, the appetite and the activity, depending on the state of the disease, could be either increased or decreased. And hormonal and suicide risk factor is one of the important characteristics of the disease, which therefore gives us a very important tool to understand the cellular and molecular mechanisms of these circadian rhythms. And the circadian rhythm is derived, the word circadian is derived from two Latin words called circa, meaning around, and diem, meaning like day. So circadian then means like approximately 1 day. Circadian rhythm basically therefore means any biological process that displays an endogenous and entrainable oscillations of about 24 hours. And this circadian rhythm is generated by a central circadian clock, which in our body is in the brain, the SCN, which is a cluster of around 10,000 neurons based in the hypothalamus. And this SCN is just placed on the top of the optic chiasma, which receives light directly from, the retina, and thereby it entrains the SCN cells, which in turn regulates and influences different activities in our body, like the sleep-wake cycle, the body, and the appetite, hormone, and the neurotransmitter release. So besides the SCN, which is the central pacemaker, there are additional peripheral clocks which have been found in most of the cells of the body. And the circadian clock is genetically encoded. This basically consists of this main primary loop, which BML1 and the clock are the main transcriptional activators, which binds to the EBOX promoter region of the period and the cryptochrome, which are abbreviated as PER and the CRIs. These PERs and the CRIs then return into the cytoplasm, where they translate into proteins for PERs and the CRIs. And over a period of 24 hours, they dimerize and go back into the nucleus, where they negatively inhibit their own activators. So over a period of 24 hours, this transcriptional translational feedback loop provides a 24-hour mechanism whereby every cell maintains the circadian clock. Besides the primary loop, there is also a secondary loop called the reverb alpha and the RORs, which also provides feedbacks into the main loop, and together they maintain the overall circadian clock in the cellular system. So how do we study the cellular rhythms in human cells? We start with the skin biopsies from the patient fibroblast cells, which are then expanded and grown in the labs. These fibroblast cells are then virally infected with a reporter construct for PER2 luciferase, whereby the luciferase indirectly provides the activity of the PER2 bioluminescence activity in the cells. So here you see an example of the on phase and the off phase and the on phase in the cells, where the bioluminescence activity over a period of 24 hours gives the luminescence, and then this bioluminescence activity is then plotted into a sine wave graph, which gives the circadian oscillations over on the recorded on a period of number of days. And from this graph, we could actually calculate the period, which means the time taken in hours to complete one oscillation, and then the amplitude, which is the counts per second measured from the baseline of the bioluminescence. And also similarly, the phase, which can be calculated for the origin of the circadian rhythm. So previous work in our lab by Dr. McCarthy and Dr. Wells have already studied in patient fibroblasts, and they could already see that compared to the controls, the bipolar patient fibroblasts had a longer circadian period. In addition, the longer circadian period and the phase delay was associated only in the lithium non-responsive patient-derived fibroblasts. In a temperature-entrained conditions, only the bipolar-derived fibroblasts showed lower amplitude, and on addition of lithium, only the bipolar-derived fibroblasts showed a lower amplitude. And in terms of finding a mechanism, they could also find that an important genetic risk factor called the CACNEC1C, which is an L-type calcium channel, and the ERK signaling pathway could be used to underline some of the mechanisms of these circadian rhythm disruptions in the fibroblasts. And to also give you an example of the different animal models which have been generated, but not to go in detail in the interest of the time, that the genetic mutations in the clock genes have been already shown that once these mutations are affected, we could already see disturbances in the mouse models where they could either mimic either the manic or the depression phase, but not like together. And these are actually the limitations which bring us to the main inferences. For example, like in the animal models, they do not express the full behavioral phenotype, for example, the suicidal phenotype, which is very, very common in those patients. And similarly, the developmental trajectory is absent in these genetic mouse models because the disease happens within a period of just few weeks and months, whereas in the patients, this happens over a period of 18 to 25 years. And similarly, all these mouse models only mimic like only one aspect, either the manic or the depression phase of the disease. Similarly, the peripheral cells using the fibroblast cells, they do not express any major neurotransmitters, for example, the glutamate or the dopamine. And similarly, they also do not have any synaptic network as it happens in the brain. And we know that the disease is actually a neuropsychiatric disease, so everything has to be related with the neurons. And similarly, since these do not have any neurotransmitters or any neuropsychiatry, there is a very key, or it lacks a very key molecular targets which could be used for underlining the therapeutic drug screening processes. So we used an iPSCs-based experimental paradigm. The idea is actually to use the patients which have been clinically diagnosed and also control subjects which are age and sex matched to use those fibroblast cells to reprogram back into human iPS stem cells using the 4 Yamanaka factors which have been already well established. And then these patient-derived iPSCs and control iPSCs could be differentiated into different kinds of neurons. In our lab, we generate into cortical neurons, so we use FGF2-based protocol. And based on these early neuronal progenitors and the differentiated neurons, we could study the circadian rhythms, the gene expression, and also the pharmacological modulation in our patient-derived cells. And we hypothesized that our neuronal cell model faithfully models the psychiatric disease phenotypes which are present in the patients. And similarly, the neural progenitor cells and the differentiated neurons, these exhibit the disturbed circadian rhythms which we have seen in the patient fibroblasts. So the differentiation protocol that we use for generating the neurons is this embryo-based Rosettes protocol, where we start with human iPSCs, where they grow as tight boundary of cells with a distinct morphology, and using directed them into like 1 to 2 weeks protocol into neural induction medium, where they first grow as free-floating brain ball cells, and then these brain ball cells, when plated again into polyornithine laminine surface, they attach, whereby they form these donut-like structures, which are actually called the neural rosettes, a resemblance of the neural tube-like structures. These neural rosettes are a rich abundance of the neural progenitor cells, which when disintegrated into single cells, can be proliferated and expanded in cell culture lab for several weeks and passages. And these NPCs could then be used to generate or enrich specific type of neurons. For example, in our lab, we used to generate cortical neurons where we differentiate them into cortical neurons, glutamatergic neurons. And this is just to basically characterize the different stages of those cells, not to go into detail, but these are the different markers. And so the first thing that we started was by looking at the gene expression profile. And this is the profile of the gene expression, where I would explain like this is the 6 hours of interval, we collected the RNA, and then we looked at the PER2 expression, which was significantly higher in the patients, whereas the Cry1, BML1, and the clock was not significant in the patient cells. We then analyzed the luminometer data by looking at the population of the cells, where we could find that compared with the control cells, the bipolar disorder, the amplitude was significantly decreased. And this is a characteristic plot of one control and patient cells, and you could appreciate from the plot here that the control cells have a very beautiful rhythms which are robust, whereas in the patient cells, it's hardly any rhythms which also dampen very fast. And then we also used a single cell image analysis to look at the circadian rhythm profiles in individual cells. This provides advantages compared with the population-based studies where we have only limited number of experiments. Here we can use a high statistical number of cells to also validate the circadian rhythm activities in different cells. And this is also a summary of the data. Compared with the control cells, we could find that the strength of the rhythmicity in our patient-derived bipolar NPCs was significantly reduced. And this is, again, a representative plot where the control have beautiful rhythms, whereas the patients have very, very weak rhythm. And just to walk you through some of our recent data from the neurons, where we again looked at the gene expression profile at 6 hours of intervals, and what we could find is now they have a beautiful circadian oscillation, which also suggests that in the mature neurons, the clock genes and the circadian clock gets more established compared to the NPCs. And again, we could see that along with the PER2, which was highly expressed in our patient cells, we also found now the Cry1 and the BML1 in the patient-derived neurons, which were significantly higher. And also we now looked at the luminometer data where we looked at the population of the cells. We could actually reproduce the same result that we found in the NPCs, that the amplitude in those patient-derived cells were significantly lower. And then when we plotted the circadian rhythms, we could find that the patient-derived neurons were completely flat only within like 2 to 3 days, whereas the control neurons actually could sustain the rhythms for at least 4 to 5 days. To summarize the data that I have shown is that compared with the patient-derived and control-derived NPCs, we could already find that the gene, which is the negative regulator or the PER2, was significantly highly expressed in the bipolar disorder. And in cell population studies, the amplitude was lower in the bipolar disorders. Similarly, the rhythms in single cells are weaker. And compared with that, when we looked at the neurons, we could additionally find PER2, Cry, and the BML1s also significantly expressed, and the rhythms, the amplitude was much weaker and dampened in the cells. So we want to continue this experiment with our neurons with additional lines, and also want to check the effect of lithium, how this could be able to modulate some of the rhythm profiles in our patient cells. And finally, I would like to acknowledge all my lab members who have helped us to generate those iPSCs into neurons, and generate those circadian rhythm profiles, and also to collaborator Dr. David Wells for giving us the facility to use the single cell imaging analysis, Dr. John Kelso for providing us the early impetus for iPSC's work and providing all the patient-related information, and Dr. Gage for giving us the protocols for iPSC's neurons. And I would be happy to take any questions. Thank you.
Very nice talk. My question relates to the states of the status of the cell after reprogramming, like after this rejuvenating process from skin cells to stem cell, and then the differentiation process from stem cell to neurons. Are you worried that some of the molecular mechanism is completely washed out during this rejuvenating process? That might impact in the finding of the right molecular changes that affect these bipolar cells versus another versus controls. And I'm glad that you see the changes are still in terms of oscillations are the same, but I'm talking about other key molecular players if in the future you're looking for them. And the second question is, are you planning to differentiate those neurons into subtypes, like dopaminergic subtypes? Marchetto was successfully able to achieve those dopaminergic neurons. So I'm wondering if in the future you're planning to compare different neurotransmitter expressing cells.
Yeah, I think both questions are really interesting. Coming to the first question regarding how rejuvenation and changes in the properties might happen during differentiation and also like during reprogramming. Yes, this is actually one of the important factors which is like considered in this field, and that's why there are like additional ways to directly reprogram from fibroblasts into neurons so that you don't have to go through this rejuvenation process and thereby unmasking or kind of inhibiting some of the processes. But the problem with the direct reprogramming is the efficiency still is pretty low. And the only experiment that could be possible with those low percentage of the cells and neurons which you get could only be used to extract some RNA to do some RNA-seq and not used for these kind of functional studies where we need like several hundred and thousands of populations. But yes, it would be also good to compare side by side if we miss any of the phenotypes during this reprogramming and differentiation. And coming to the other part, again, like differentiating into subtypes, yes, that's actually also one of our goal. We wanted to start with one of the basic type of neurons which are easy to grow, and then where we can first establish that the system is working, and we could recapitulate the phenotype that we got in the fibroblast. But yes, in the coming weeks and like years, we are planning to differentiate also in other subtype, and then see whether the difference is still there or it's more different in other subtype of neurons.
Let's thank Himanshu for a really nice talk again. Our next speaker is Bosi Chen. She's here. Okay. She's a second-year doctoral student at the SDSU UC San Diego Joint Doctoral Program in Clinical Psychology, and is mentored by Dr. Ina Fishman and Dr. Marty Serino at SDSU. Her research interest is in early brain development of autism with a focus on resting state functional connectivity. She graduated from New York University with a BA in Psychology and Economics, and also worked as a research associate at the NYU Child Study Center to study the underlying neural mechanisms in autism spectrum disorder and ADHD. And Ms. Chen will present on atypical brain network connectivity proximal to behavioral symptom onset in toddlers with autism spectrum disorders.
Thank you. Thank you for having me here. It's an honor to be able to present my work in the symposium as a trainee in the department. So today I will be mainly talking about the organization of functional networks in toddlers with autism spectrum disorders compared to typically developing peers. So autism spectrum disorders are disorders of development with behavioral signs of ASDs emerging usually in the first years of life. Previous research has shown that ASD diagnoses established as early as 18 to 24 months are relatively stable and often referred as the clinical best estimate of ASD when made before age 3. So besides the behavioral symptoms of ASDs in early development, brain abnormalities have also been identified, especially in infants at risk for ASDs based on data from the Baby Sibling Consortium and other large-scale efforts. However, little is known about the brain functional network development and organization at the developmental period proximate to symptom onsets. So our current knowledge in functional networks have been heavily informed by resting state functional connectivity, which measures the temporal correlations of spontaneous low frequency fluctuations of the brain blood oxygen level dependent signals in the brain using resting state functional resonance imaging, RFMRI. From the literature on functional network development in typical development, we know that most functional networks are present at birth, and the maturation of functional networks follow the order in which behavioral milestones are achieved, such that sensory network usually mature first, followed by attention and language network and executive networks. Yet there is less evidence available on the maturation of functional networks in infants and toddlers with ASDs. Specifically, we are interested in these two major questions. Do functional networks in toddlers with ASDs mature to the same extent as typically developing peers at this point in development? Do the functional networks show the same level of integration and segregation? So in order to explore these questions, we use data from the SDSU Toddler MRI project. It is an ongoing longitudinal studies of brain network organization and development in toddlers with ASD. The sample for this current study consists of cross-sectional data of young children with ASDs and their typically developing peers between the age of 15 and 41 months. 19 were diagnosed with ASDs and 19 were typically developing. Clinical best estimate of ASD diagnoses were established based on the DSM-5 criteria and was supported by the Autism Diagnostic Observation Schedule, which is a gold standard diagnostic tool for ASD. So as expected, our participants with ASD show a delayed development across multiple behavioral domains, including gross and fine motor skills, visual reception, receptive and expressive language and socialization skills, indexed by the Mullen Scale of Early Learning, which is a clinician-administered measure, and the Vineland Adaptive Behavioral Scale, which is a parent interview. So as you can imagine, scanning young children during sleep is not an easy task. Our team has developed a series of materials to help parents and kids become more familiar with our scanning environments, including a social story that parents can read to their kids at home, a video of what the scanning facility and the preparation steps are like. We also ask parents to practice putting in earplugs and playing MRI sounds while their kids are at home, sleeping at home for a week prior to coming to the scan. Additionally, we have different options for the children to fall asleep at the scanning site. And we also try to decorate the scanner more child-friendly so that it's easier for the child to fall asleep in there. So as you can see in this picture right here, we kind of decorated more like going into a space machine. So this is kind of a procedure that we have developed over time. So our overall success rate for getting usable fMRI data is about 65%. So all the toddlers were scanned during natural sleep at night on the 3TG scanner as CFMRI. Resting state fMRI data were collected with the acquisition parameters specified on this slide. We applied standard preprocessing pipelines to preprocess the resting state fMRI data. Because these data were collected during sleep, overall there is relatively low head motion in both the ASD and TD groups. However, head motion was not well matched between groups, with toddlers with ASD having relatively higher head motion than TD toddlers. The primary analysis was repeated with a smaller sample with closely matched motion, and then I will go into a little bit more detail later on in the talk. So with the resting state fMRI data, we ran independent component analysis, ICA, to identify resting state functional networks. So ICA is a commonly used approach for identifying functional networks. It is a data-driven approach that basically decomposes our fMRI data into a set of spatial components that are maximally independent. So 20 components were extracted, and spatial and temporal courses of these components were visually inspected and compared to the Smith template and the HCP IC components in order to distinguish the neuronal components, which we call the resting state networks, from artificial noise components, such as coming from breathing, cardiovascular, or head motion. So in the end, 12 components were retained, including 5 visual, 1 auditory, 3 sensory motor, 1 inferior parietal, 1 salience, and 1 default mode component. And here are all the retained components shown on this slide. So regions of interest were extracted from the components identified in our previous ICA step and entered into ROI to RI connectivity analysis. So we observed a general atypical connectivity patterns both within the visual network and between the visual and other networks. So after FDR corrections, there is a underconnectivity between 2 RIs within the visual network and overconnectivity between RIs in visual and sensory motor networks. And this analysis was repeated with a motion-matched subsample, and the results were essentially the same. And these results seem to suggest differences in connectivity patterns within the visual and between visual and other networks in ASD toddlers compared to typically developing peers. And we want to further understand how these different connectivity patterns are related to age and behavioral indices. So we tested the correlation between average within network connectivity and age while controlling for head motion in ASD and TD separately. So we found a significant positive correlation between connectivity within the IPC network and age in ASD toddlers. However, there's no significant age-related correlation with any other network in both ASD and TD. So these are all partial correlation plots here. So given that our findings in the RI to RI analysis implicated exclusively the connectivity differences between visual and other networks, we then go on to examine the correlations between age and connectivity between visual and other networks while controlling for head motion. So interestingly, we found significant negative correlation with age in the connectivity between visual and auditory and between visual and salience network only in our ASD toddlers. And the age by diagnostic group interactions are also significant. We observe a similar trend in the connectivity between visual and IPC and between visual and sensory motor networks, though not reaching statistical significance. However, this age-related decrease in between network connectivity is not evident in our TD participants. And additionally, we want to examine if the atypical connectivity patterns are associated with autism symptom severity. We found a positive association between autism symptom severity and connectivity between visual and auditory, visual and salience, and visual and IPC networks. So these results indicated higher between network connectivity involving the visual network is correlated with greater symptom severity indexed by the ADOS total scores. In relation to cognitive development, we observed that lower connectivity between visual and salience network is associated with better performance on the Mullen scale of early learning in children with ASDs, but not in typically developing toddlers. So these results seem to suggest that greater functional connectivity between visual and other networks, hence reduced network differentiation at this early age, is associated with more severe ASD symptoms and better and lower cognitive development. Besides examining the connectivity patterns within and between specific networks, we also examined the whole brain connectivity patterns. So whole brain connectivity maps from each IC were extracted and compared between groups. However, we found no between group differences. Some recent reports suggested that increased variability of functional connectivity across individual is perhaps the critical characteristic of the ASD brain. These results are based on findings in older school-aged children and young adults with ASDs. And it is unknown whether similar pattern exists early in life. So we calculate a correlation between every ASD toddler's connectivity map with every ASD toddler's, and the correlation between every TD toddler's with connectivity map with every other TD toddler's. So when we compare average correlation across all the ICs, we found that ASD toddlers are actually more similar to other ASD toddlers in their whole brain connectivity maps in the primary visual, inferior parietal, and salience network compared to the extent to which TD toddlers' connectivity map were similar to their TD peers. And this result suggests the more variable functional connectivity patterns observed in school-age children and adults with ASD are not really evident in this early developmental period. So to sum up all the findings from this study, we found atypical functional connectivity implicating visual network in 1.5 to 3.5-year-old toddlers with ASDs, including weaker connectivity within visual network and stronger connectivity between visual and motor regions. We also identified age-related effects on the differentiation or between network connectivity between visual network and auditory and salience network, such that toddlers with ASDs show decline in these between network connections between age 1.5 and 3.5 years, where no such age effect was detected in TD children. And we interpreted this finding as probably a maturational delay in network segregation, which is the between, for example, between primary sensory network and salience network in ASDs. The interpretation was supported by the data indicating that greater network segregation in toddlers with ASDs were associated with decreased symptoms and greater performance on measures of cognitive development. Finally, in contrast to previous findings of greater variability in functional connectivity patterns in school-age children and young adults with ASD, toddlers with ASD show more similar functional connectivity patterns in primary visual, inferior parietal, and salience network compared to their TD peers. So our current study is limited by the moderate sample size. However, as data acquisition is still ongoing, we expect to repeat the analysis as our sample size grows. Additionally, the longitudinal design of the study, the longitudinal analysis of functional network development will be conducted to compare functional network development trajectories in ASD compared to TD. We will also examine the relationship between functional connectivity indices and changes in symptom severity using our longitudinal data available. All right, so just to wrap up, I would like to thank the participants and their families and members of the Brain Development Imaging Laboratory for the support. I would also like to thank the funding source and all of your attention.
Very nice work. Were you surprised that most of the findings were with the visual network, given that visual problems aren't really very prominent in ASD, and in fact, in your own data, the visual perception domain was kind of one of the least affected. Right. And sort of how do you interpret that sort of disconnect? And did you look at whether there was a correlation with visual perception domain specifically and the findings that you had in those networks?
Thank you for the question. Yeah, so when I first did analysis to extract those components, I wasn't expecting a visual network as the one that showed all the differences. But you know, in the ASD literature, in the development in these kids, we know that there are some visual stimming behaviors that are quite common. But in terms of their visual receptive scores on the Mullen, that's the one that usually show the least difference between ASD and TDs. And one other thing I'm thinking about is maybe the tasks that were in the visual reception task is not maybe super relevant to what we think about as visual abnormalities in terms of maybe visual stimming and other more sensory-driven processes. So that's definitely something that we were not expecting going into the analysis, but it's somehow become the strongest finding that we have. Thank you for the question.
Using anatomic MRI instead of functional in ASD populations of this age, the way that the girls with autism's brains are different than typical girls is different in a different way than boys with autism, age by sex by diagnosis of action. Basically that in girls there is relatively greater white matter and in boys less. So that, with the sample size you wouldn't be able to statistically see a difference unless it was greater than like 15, 20%. So it's likely it's just, you can't answer this directly, but is there any suggestion of a male-female difference in this data in terms of particularly with like the variability of the networks, which also in just typical people, the variability is much higher in males than females?
Great question. We haven't done any gender analysis in our sample because, as you can tell, that in our participants we only have about like 9 female in each group, so we were not really able to do any gender analysis. But that's a really interesting question, whether girls with ASD's brain connectivity is different. It's different from boys with ASD. If we have more data, I would definitely want to explore that question a little bit more. But with our current data set, we were not able to do any analysis with that.
Okay, let's thank Rosie once again. And our next speaker is Dr. Megha Amar. She obtained her PhD in India, and her dissertation elaborated the role of REM sleep and noradrenaline in maintaining neuronal excitability. And she moved to the United States in 2015 and investigated the role of sleep in synaptic homeostasis using the Drosophila model system. And she did this work at USC. She came to the Department of Psychiatry in 2016, and she joined Dr. Lilia Iakachev's lab in our department. Here, her research has focused on investigating the molecular mechanisms of autism spectrum disorders using a mouse model system. And she will today present her work on autism-associated germline Choline-3 mutations that severely impact mouse brain development.
Thank you so much, Dr. Mishra, for the kind introduction. And I would like to extend my thanks to the organizing committee to giving me this opportunity, and it's an honor to present my research in the symposium. So the title of my talk is Autism-Associated Germline Choline-3 Mutations Severely Impact Mouse Brain Development. And Bosi already gave a brief introduction about autism. So autism is not a single condition. It is, there are several disorders which are under the umbrella of autism, and the individuals with autism have social impairment, they have language deficits and restricted or repetitive interest. And it is a spectrum, means there are milder forms and there are more severe forms of the autism. And in addition to these 3 core symptoms, the spectrum can include anxiety, attention deficit hyperactivity disorders, or also the intellectual disability. It is one of the fastest growing neurodevelopmental condition. According to CDC 2018 report, 1 in 59 US individuals are affected by autism, and boys have 4 times more risk for autism than girls. And despite of its high prevalence rate, the molecular pathways involved in the disease is still largely uncharacterized. Though the incremental advances in the discovery of new genes associated with autism risk are already enhancing the clinical definitions and the diagnostic tool. However, there is no reliable biomarker for the disease, and most of the available treatments are symptom-based, there is no drug available for the core deficits of autism. Therefore, the long-term goal of our lab is to identify the molecular pathways disrupted by the genetic mutations in autism and target those with drugs. So one of the gene of interest for our lab is the Choline 3. So Choline 3 is one of 28 high-risk autism risk gene with genome-wide FDR 1%. In total, 19 rare mutation has been identified in the cases with autism, intellectual disability, developmental delay, and schizophrenia, while 0 in controls. And what Choline 3 is, this protein is a E3 ubiquitin ligase, which mediates ubiquitination and subsequent degradation of the target proteins. It is essential for the embryonic development, as complete deletion of this gene leads to the embryonic lethality. It regulates diverse cellular functions such as cell cycle regulation, stress response, and protein trafficking. But how Choline 3 mutation lead to the autism is still not clear. So the hypothesis was that Choline 3 influenced brain development by the ROA pathway. So the recent study from our lab, by using the spatiotemporal developmental brain gene expression data combined with the protein-protein interaction network, demonstrated that one of the interacting partner for the Choline 3 is the KCTD13. And KCTD13 is the part of 16p11.2 CNV. And I would like to add here the 16p11.2 copy number variation is the most common and high risk copy number variation in autism and other psychiatric diseases. Both deletion and duplication in the CNV leads to the autism and other psychiatric diseases, and the children with the CNV mutations have macrocephaly and microcephaly phenotype. So it was shown that one of the interacting partners for the Choline 3 is the KCTD13, and this complex regulates the ubiquitination and subsequent degradation of the small GTPase ROA. And ROA is a major regulator of the actin cytoskeletal and neuronal growth and expansion in early cortical development. So it was proposed that deletion in the 16p11.2 or de novo loss of function mutation in the Choline 3 leads to accumulation of ROA in the brain, which in turn results in aberrant neuronal growth and expansion in early cortical development. So in order to validate this hypothesis and to understand how Choline 3 mutation affect the brain development, we generated the Choline 3 mouse model using CRISPR-Cas9 genome editing technology. We did the behavior phenotype for the AS relevant behavior, and we investigated the brain abnormalities using the MRI. Next, to understand the molecular pathway disrupted by the Choline 3 mutation, we did the transcriptomic and proteomic profiling of the Choline 3 mouse model, and we have a long-term goal to use drug treatment for reversing the phenotypes. So coming on to our first objective, for generation of the Choline 3 mutant mouse model, we targeted our guide RNA towards the exon 6, which contained one ASD-associated de novo loss of function mutation, and confirmation of the mutation was done by the Sanger sequencing and the T cloning. So here this is the guide RNA sequence, and the mutation is insertion of the mutation was confirmed. As you can see over here, there is a one nucleotide insertion in the sequence leading to the frame shift mutation. Further, the insertion of mutation was validated at the protein and gene level. The Choline 3 level is significantly reduced in the Choline 3 mice as compared to the GAPDH. And at the gene level, we did the quantitative real-time PCR, and we observed significant reduction in the transcript level of the Choline 3. It suggests that the frame shift transcripts underwent degradation. Next, we did the behavior phenotype for the ASD relevant behaviors, specifically cognitive and the social behaviors. So first, we observed that these mice are underweight. Both male and female Choline 3 mice have the reduced weight as compared to the wild-type mice. And in the open field, to measure the anxiety, exploratory behavior, and the locomotion defects in these mice, we observed that there is no difference in the center versus periphery for the Choline 3 mice. However, the total distance traveled and the total speed is higher for the Choline 3 mice as compared to the wild type mice. It suggests that these mice are hyperactive. Next, to study the cognitive impairment, we did novel object recognition test. So the idea is we give the mouse the identical object and during the training session, and in the test we replace one of the familiar object with the novel object, and we calculate or measure the time the mouse spent with the novel object or the familiar object. And we observed here that the preference for the novel object is significantly reduced for the Choline 3 mice as compared to the wild type mice. And for studying the social interaction, we did a three-chamber test in the two phases. The first phase was the sociability, where the mouse was given one novel mouse, and in the other chamber we put the novel object, and we measure the time spent in each chamber with the novel object or the novel mouse. In normal condition, mouse should spend more time with the novel mouse as compared to the novel object. And what we observe, both Choline 3 and the wild type mice spend more time with the novel mouse as compared to the novel object, but there is a significant reduction in the interaction with the novel mouse for the Choline 3. In the second phase, which is a social novelty phase, here we put one familiar mouse in one chamber, and in the other chamber we put the novel mouse, and we measure the time spent in each chamber with the novel or familiar mouse. So here we observe that Choline 3 mouse was not able to distinguish between the novel or the familiar mouse. So these mice have a reduced social drive, and also they demonstrate the cognitive impairment. Next, to study the brain abnormalities in these mice, we did the MRI on 8 to 10 weeks old adult mice brain, and this was done in the collaboration with the Jason Lurch lab. So the blue regions represent the significantly reduced relative brain regions with FDR 5%, while the subcortical red regions are the significantly larger regions. So here we observe that total brain volume for the Choline 3 mice are significantly reduced as compared to the wild type mice, and the gray matter volume, which represent the neuronal cell bodies, are also significantly reduced in both relative and the absolute volume for the Choline 3 mice. There is also marginal difference in the cortical thickness around the regions of the somatosensory cortex, visual cortex, and entorhinal cortex, but we will be further validating this result with the cortical layer staining. And almost 46% of the brain regions are significantly altered at FDR 5%. So regions such as entorhinal cortex, somatosensory cortex, cerebellums are significantly reduced, while hypothalamus, thalamus, and the midbrains are significantly increased at FDR 5% in these mice. So to understand what are the molecular pathways and how the Choline 3 mutation affect the other gene expression, we did the transcriptomic and proteomic profiling of the Choline 3 mice. We did it at the developmental trajectory starting from the embryonic day E17.5 and postnatal day 7 and the adult to capture the specific changes during the developmental period. So what we observe here, so this is the volcano plot from the embryonic day E17.5 cerebral cortex. The yellow dot represents the significantly upregulated genes, while the blue dots represent the significantly downregulated genes at FDR 10%. As you can see here, the Choline 3 is the most significant downregulated genes in the Choline 3 mice. And next we run the Go term analysis to see what these genes, differentially expressed genes, are enriched in. And we observe that these genes are enriched in the biological processes such as exogenesis, cytoskeletal organization, and the cell migration. And this molecular pathways are disrupted not only in the embryonic day E17, but also in the adult mouse brain, cerebral cortex. So in order to validate these molecular pathways, we did the cortical neuronal culture at the same developmental time period, E17.5, and after 7 days of culture, we measured the neuroid length in the cultured neurons with the MAP2 staining, and we observed that total neuroid length was significantly reduced for the Choline 3 mice. So these mice have disrupted neuronal growth and expansion pathways. And next, we did the proteomics to study the impact of the mutation on other proteins. And after doing the proteomics, we did perform the weighted gene co-expression network analysis on differentially expressed protein to identify modules of co-expressed proteins across all samples. Each module was then tested for their association with the genotype. And this blue module over here, it is significantly upregulated in the Choline 3 mice as tested by the linear regression model. And the trend is also clear on the eigen gene expression plot that this is upregulated in all different samples of the Choline 3 mice. So the network plot shows the hub genes from these module, and we can see over here in the orange that these proteins are upregulated in the Choline 3 mutant mice. So next we did the functional enrichment analysis by the G-profiler to see what these proteins are enriched in. And we observed that these proteins are enriched in the Rho GTPase, negative regulation of the cell motility and cell migration. So it shows that possibility of the Rho pathway in the regulation of the phenotype in the Choline 3 mice. So in order to validate this module and potential dysregulation of the Rho in the Choline 3 mice, we did the Western blotting from the cerebral cortex of the Choline 3 mice from the adult brain, and we observed significant increase in the total Rho level in the Choline 3 mice as compared to the wild type mice. And so the active form of the Rho is the GTP bound form, and we wanted to check whether the accumulated form of the Rho is the active or functional form or the inactive form. So we did the immunoprecipitation with a specific bead which can only capture the GTP bound Rho, and we observed that active Rho is actually accumulated in the Choline 3 mice, which suggests the potential dysregulation of the Rho pathway in these Choline 3 mice. So to summarize, what we observe here is the Choline 3 mice are smaller and they have severe brain abnormalities, and the behavior data shows that these mice are hyperactive and have cognitive impairment and the social drive. And Choline 3 mutation affect and dysregulate other genes and molecular pathways such as neuronal growth and migration, and we implicate the Rho pathway at least for the cerebral cortex. So to acknowledge, I would like to acknowledge my lab. And for the behavior setup, I would like to acknowledge NEAT lab, Dr. Namanathan and the Puneet. For the imaging, I would like to acknowledge Jason Lurch lab in Canada, and we did proteomics with the Yates lab and our other collaborator, Dr. Motri and the Jonathan Sibeth. And for the funding, I would like to thank NIMH and the Safari Foundation. And thank you so much, all of you. And I would like to take any questions now. Thank you.
Okay, our next speaker is Karen Arabanovich. She is a second-year doctoral student in the SDSU UCSD Joint Doctoral Program in Clinical Psychology, and she's mentored by Dr. Cinnamon Bloss. Karen's research focuses on the ethical, legal, and social implications of emerging technologies in clinical care, as well as the societal level impact of technology more generally. More recently, she has been studying privacy and is working with Dr. Bloss to develop a reliable and valid self-report measure of privacy attitudes and behaviors in the context of health information sharing. Karen received her master's in narrative medicine from Columbia University, and where she studied clinician-patient relationships, and her AB in anthropology from Washington University in St. Louis. Ms. Rabanovich will present on privacy behaviors, a predictive of suicidality and high-risk behavior in physicians, nurses, and students. Please welcome Karen.
Good morning. My name is Karen Rabanovich. I'm a clinical psychology doctoral student who's driven to understand how we can better meet the mental health needs of healthcare providers. I've called this talk Honoring Privacy, Identifying and Responding to Healthcare Providers' Mental Health Needs. I hope to weave together a compelling story for you with two threads: These are threads I think a lot about: privacy, specifically privacy behaviors, and mental health, specifically the mental health of healthcare providers. As many of you may be aware of, there's a growing concern about the mental health of our healthcare providers in domains like depression, suicide, alcohol abuse. Healthcare providers may have privacy concerns in accessing and adopting mental health care due to things like stigma, discrimination, and licensing concerns. I had an opportunity to test out some of my ideas around privacy behaviors and how they might be connected with mental health in providers. I had an opportunity to work with a set of data collected through the HERE program here at UCSD. This is data that is from an online screening tool, a digital health tool, meant to connect UCSD providers with mental health resources and support. My take-home message for all of you today is two things. One, that privacy behaviors may have clinical correlates, and that honoring privacy could help us intervene and get mental health resources to those who are most in need. Briefly, my outline: I'll first talk about privacy behaviors, then get into the HEALER Education and Assessment and Referral Program, the HERE program. I'll weave together the two threads. I'll summarize, talk about future directions based on this work, and I'll end with acknowledgements and room for questions. Why does this matter? Digital health is an expanding frontier for mental health support and treatment. Indeed, I think many like myself are very excited because digital health allows us to overcome structural barriers and extend the reach of mental health care. At the same time, there are privacy concerns that limit the adoption of these online resources and technologies. Studies suggest that privacy concerns are barriers to utilizing digital health. And at the same time, physician, nurse, trainee suicide is a growing concern. And there are major barriers to help seeking and receiving care due to stigma and fear. As the statistic on the right shares, 400 US physicians take their lives every year. To put it another way, that's 3 UC San Diego medical school classes gone every year. I spent a lot of time alongside Dr. Simon Bloss in our wonderful lab thinking about privacy attitudes and behaviors. What are they? And how do we measure them? There's a lack of reliable and valid tools out there in the literature to help measure these privacy attitudes and behaviors. And there seems to be a growing interest in privacy nowadays. And so our main objective is developing a psychometrically sound instrument to better capture privacy attitudes and behaviors. I've been spearheading the quantitative analyses of this project, helping us ensure that we're following a rigorous methodology in developing this instrument. And I've been thinking a lot about what are different types of privacy behaviors? How can we validate this instrument? One idea I had was using non-response to demographic items as a type of privacy behavior. And it's this idea that I took and tried out with the HERE data set. Very briefly, the HERE program here at UCSD was created in response to a UCSD physician and faculty member who took his life several years ago. It was that, coupled with startling rates of depression, suicide, and alcohol abuse here amongst UCSD providers, that really moved the HERE program to be created. And so it offers various resources for our UCSD community. One of those resources is an online digital health tool or a screening tool for UCSD providers and affiliates. It's a completely voluntary screener, and users can access individualized support completely anonymously if they so choose. And so it was with this data set, I looked at data from January to October of 2018 with an N of 571 UCSD providers, students, and affiliates. And I looked at things like depression, suicidality, functional impairment from alcohol abuse, and stigma in seeking mental health care services. So how is the privacy behavior operationalized? As I already mentioned, my assumption is that non-response to a demographic item is a type of privacy behavior. And so the privacy behavior count was the number of missing responses or prefer not to answers to demographic items, such that a higher count was indicative of someone trying to maintain more privacy. There were 6 demographic items: gender, race, age, position, department, and class. I tabulated privacy behaviors in a couple of steps. First, if a responder skipped position, gender, race, age, that was automatically 1 point per missing item. If a responder chose prefer not to answer to any of the demographic items, that was also 1 point per missing item. And then, depending on what position someone identified, I used that and some branching logic to then go ahead and determine whether missing class or department was indeed a missing item towards the privacy behavior count. And the way that the screener looks for the user is that it asks if someone is a student or resident to go ahead and fill out their class or what year they are. And it asks that if someone is a resident, fellow, or faculty, to go ahead and indicate which department they're a part of. So getting into some of the results. First, depression. So this was measured with the PHQ-9, so looking at depressive symptoms over the last 2 weeks. And the average was 8.94, which falls in the mild depression range, but over 42% of the sample indicated at least moderate levels of depression. When it comes to suicidality in the past 2 weeks, almost 14% reported having thoughts of being better off dead, per the number 9 item on the PHQ-9. 8.8% reported having thoughts about taking their own life, called suicidal thoughts. 4.2% reported planning ways to take their own life, suicidal plans. And about 1%, or 7 individuals in our community, reported having done things to hurt themselves, or suicidal actions. There was about 7% reported an attempted suicide across their lifetime. Over 21% reported worrying about stigma for seeking mental health care services, and about 5.5% reported work or school functional impairment from alcohol use. Getting into the privacy behavior count, so on the left I've got a distribution. Just under a quarter of the sample had a privacy behavior count of 1 or more. Of those individuals, on average, 1 to 2 items were missing. And on the right, I've got a table with a breakdown of the non-response items. So the most frequently skipped was age, followed very closely by department, and then in third, race and class, and then position and gender. I went ahead and ran some regression models with the privacy behavior count used as a single independent variable. While it was not statistically significant in its association with the total depression scores, it was statistically significant in its association with suicidality, as measured by the 9th item on the PHQ-9, suicidal thoughts, plans, and actions. In other words, the higher the privacy behavior count, the more severe the suicidality. It was not statistically significant in predicting suicidal attempts across the lifetime. It was statistically significant in its association with functional impairment in work or school due to drinking, and also worrying about stigma for seeking mental health care services. So again, the higher the privacy behavior count, the worse the functional impairment, and someone indicating worrying about stigma for seeking mental health care services. So to summarize the findings, over 42% indicate at least moderate depression, over 21% worried about the stigma in seeking mental health care, Almost 14% had thoughts of being better off dead in the last 2 weeks, and about 5.5% reported work school functional impairment from alcohol use. Just under a quarter maintained their privacy and reporting, and age and department were the most frequently skipped items. Privacy was significantly associated with suicidality, work school performance, and stigma in seeking services. Non-reporting may be an important clinical correlate for us to think about moving forward. And so future directions, I'm really eager to dig into some other data sets and see if these findings are replicated. I also want to offer this method of counting non-response. It may be a generalizable technique for others to use with survey data, and the good news is we can use it on existing data sets, anything that might have demographic items. When it comes to digital health, I think a lot about now how we might integrate privacy behaviors into the design, development, and deployment of interventions. How can we test the adoption of, engagement with, and benefit from digital health as a function of privacy behaviors? Give people the choice. Perhaps they don't need to identify themselves entirely in order to benefit from our digital health tools. And then the last cautionary tale is the risk of asking too much personal information of users. We live in a day and age in which more data seems to be better, but at what cost? Perhaps we're risking deterring individuals from engaging with our tools. And so asking too much personal information may be a barrier to reaching those who need care the most. So it's certainly an important consideration when developing online and digital health support. I'd like to acknowledge my wonderful mentor, Dr. Cinnamon Bloss, in our lab, Dr. Sid Zusuk and the HERE program. I want to thank the selection committee of the Lewis L. Judd Young Investigator Symposium for giving me this opportunity to share this important work with all of you. I'd like to thank the JDP Clinical Psych program, my amazing classmates, faculty, and staff. And for us to remember, this is our community. These are our colleagues, our peers, our students, our teachers, and their mental health matters. Thank you for your attention. I'm happy to answer any questions.
Thank you. Hello? Okay. So how do you know that this is about privacy as much as it could be about other things like, somebody not being able to answer, not wanting to answer race or gender because they're sort of mixed, or because they're in a small class or they're in a small department, and they, by saying some of these things, it would be much more easy to identify themselves. So is it really about privacy, or is it really that these people that are not answering questions are just in a really high-risk group, and they're just trying to prevent themselves from being more identified, or the survey just doesn't really have options for them, and that they're sort of already socially isolated from their peers in this way that they don't really fit into as many categories as the people who are more easily able to answer some of those questions.
So maybe, so the question is about how do we know this is a type of privacy? Perhaps these are individuals who are already stigmatized and may not want to disclose information. Is that right?
Yeah, yeah.
Yeah, I mean, I think that perhaps we can think about information sharing as a type of, perhaps it goes into the realm of privacy. Again, this is just an assumption that I'm having leading into it. Nevertheless, I think that whichever way you want to cut it, that non-response, rather than being a nuisance, might actually be an important metric for us to follow. So there's certainly more work to be done. Thank you.
Any more questions?
did you look into trainees versus faculty and staff in terms of, I'm thinking in terms of you can see some of the association of workload, like if it's something that's part of the university where we could build, or if we could build some sort of initiatives to help trainees perhaps when it comes to mental health. Is it the work burden, or is it something outside of people's academic lives?
Yeah, so whether there are kind of differences between the various groups and that being a way to tailor or better understand those differences. Yeah, I mean, I'm totally game with that. I think that could be definitely a next step. I didn't look at differences between the different positions. So that's certainly a next step. And I think that it's important for us to tailor these mental health resources to meeting people where they're at. So whether they're a trainee or a faculty member or a nurse, they may have very different needs. So, yeah.
I don't have a, am I on? Yes. I don't have a question, just I wanted to thank you for this important work and to thank Sid and the members of the HERE committee. This is some of the most important work that we could do in our departments and across the medical school, across the JDP. Each year we do lose students here at UCSD, so really stay strong, keep up the work, and keep digging into these databases and help us reduce the loss of our best.
I will. Thank you.
Thank you, Karen, for a very important talk. We're taking a 5-minute break.
Check 1, check 2. 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 then for yours, do you want presenter view? It's actually running off of this keyboard. I have a timer right here, so I'll just kind of keep that like that. Just kind of like that, and then here. And then, yeah, so your advance. Actually, I'll just call it. Nothing. Check one, check two. Check one, check two. And then really just like speak right into it like that.
Oh, okay.
If you're like this, it's not going to pick it up. If you're like this, it's going to pick it up. It would be this mouse. Okay, just look at this guy.
Oh, right, right.
You don't even have to.
Very good. Okay.
Yeah, however you want it.
Yeah,
That's good.
I think.
Why don't we all start to take our seats again? Got some great speakers for the second half of the session here, as we did in the first. Come on in.
Hey everyone, thanks for joining for a very exciting second half like the first. Our next speaker is Dr. Matthew Kline. He's currently a third-year resident in our psychiatry research track program. He obtained his MD PhD from Albert Einstein College of Medicine, where he studied the synaptic pathology of autism spectrum disorders. He was awarded an F31 grant from the NINDS for his predoctoral research. Currently, he is mentored by Dr. Robert Malinow in the neuroscience department. He has won several recent awards, including the Maja Memorial Research Award, the APA Research Colloquium Award, the SOBP Annual Meeting Research Award, and the NIMH Outstanding Resident Award. Dr. Kline will present on the antidepressant effects of ketamine on lateral habenula activity and whether it requires endogenous opioid signaling. Thank you. Please welcome Matthew.
Thank you. Thank you to the committee for giving me the opportunity to come here today and share my work looking at the synaptic mechanisms behind the antidepressive effects of ketamine. And thank you all for coming here and listening. Ketamine was first discovered in the 60s. It's been used as an anesthetic agent
while. But for the past 20 years, there's been growing interest in the antidepressant properties of ketamine. Now, there's a couple things that make ketamine quite interesting. The first probably being the rapidity of its antidepressive effects. In some studies, up to 60% of patients showed a response, a decrease in their depressive symptoms in just 24 hours after a single infusion of ketamine. And after multiple infusions, up to 40% of patients can see remission of their symptoms in just 7 days. And this is a lot quicker than the weeks to months that our typical antidepressants take. It also has been shown to reduce acute suicidality, and it was just recently approved for treatment-resistant depression, TRD, by the FDA a couple weeks ago, intranasal version. So ketamine, this parent compound shown here on the left, is ostensibly an NMDA receptor antagonist. But it's actually metabolized quite quickly in the body, and its metabolites show activity at a wide variety of different receptors. So much so that this is actually from the product insert of the intranasal ketamine, that they admit that by the mechanism by which ketamine exerts its antidepressive effects is currently unknown. So this is a good research question. It's not, doesn't seem to be an SSRI, a serotonin reuptake inhibitor, like our current pharmacology. How is ketamine producing this antidepressive effect? So a group had a study a couple months ago, a group out of Stanford, I think, that caused quite a stir, suggesting that ketamine is actually acting as an opiate. Ketamine is binding to opiate receptors, and that's how it's causing its antidepressive effect. And what they did is they gave a small group of patients a single dose of IV ketamine, and an X here are days, or time in days post-ketamine. The y-axis is a depressive index, where 0 being no symptoms and higher scores being more severe symptoms. And so in green here, this group of patients got their dose of ketamine and showed a pretty rapid reduction in depressive symptoms that lasted for about 14 days. They took another group of patients, shown here in orange, but now they pre-treated these patients with naltrexone. Naltrexone being opiate receptor antagonist, so blocking opiate receptors. And now, as shown in this group in orange, this completely blocked the antidepressive effects of ketamine. These patients did not show any reduction in their depressive symptoms. And so they took these results, interpreted the results that ketamine is an opiate. Ketamine is binding to opiate receptors to produce its antidepressive response. And that's why naltrexone, the opiate receptor antagonist, blocks antidepressive response. And so this caused kind of a stir in articles like this. Is there nothing new under the sun? Is ketamine just an antidepressant just because it's an opiate? The field has known that opiates have antidepressant properties. They can be used for mood augmentation. And perhaps in the current climate of an opiate crisis, if we're essentially giving folks what amounts to IV opiates, might not be the best antidepressive treatment. So this is where we came in, looking at this problem. Is ketamine an opiate? How is ketamine producing its antidepressive response? Through which mechanism? And so the work I'd like to share with you today is that no, we do not think that ketamine is an opiate. I don't think that ketamine binds to opiate receptors to produce its antidepressive response. I think it is binding to NMDA receptors. However, in our rodent and cellular behavioral studies, we've shown that, that opiate activity actually is necessary for ketamine effects. You do need intact opiate receptor to get the antidepressive effects of ketamine. However, opiate activity alone, just binding to the opiate receptor, does not appear to be sufficient to produce the antidepressive effects of ketamine. And so the way we did these studies, we took a rat model of depression known as congenital learned helplessness, CLH rats. This is an inbred rat line that's been inbred to select for helplessness behavior. Basically, in times of stress, they just give up. This rat model displays depressive-like symptoms such as anhedonia, amotivation, avolition. What's nice about this model is that if you give them SSRIs, antidepressant treatments, they get better, their symptoms improve. And we think that the reason these rats display these depressive-like symptoms is because they have abnormal reward processing. There's an area of the brain known as the lateral habenula that's involved in reward processing. These animals, which I'll get into in more detail later, have hyperactivity in the habenula and just maybe never feel the reward that they expect. And that could lead to these depressive-like symptoms. So the way we test, we quantify depressive-like symptoms in this model is a forced swim test. A lot of labs use this test. We basically put a rat in a bucket of water, and on the graph on the below is a measure of movement. a rat will kind of swim around. This is a little sped up. It's not quite as frantic. It swims around trying to escape. A wild-type rat will try to escape for the entire 10 minutes it's in there. But one of these helpless rats will kind of give up pretty quickly, and you'll see that the rat will become immobile in a little bit. Right now, it kind of just floats there. It's like, well, this is my life now. I guess this is, what's happening. This sucks. There's a period of immobility. You'll see another kind of longer one. And basically what we do is we quantify the amount of time this rat spends immobile. Here's a longer period as a measure of its depression. And what's nice about this test is that it works, a variety of labs have used it in a variety of different mouse and rat models. If you kind of stress the rat more or do things that cause it to be more depressed, it becomes more immobile. If you give it antidepressants or other treatments like that, it actually reduces immobility. So there does seem to be some predictive value that the amount of immobility is some quantification of this depressive-like state. So the graph that I'm showing here, in blue, this is the activity of a single animal. In the x-axis is time in minutes, and the y-axis is a measure of movement. And so in blue, this wild-type rat, as I mentioned, will swim around the entire time trying to escape. This is a stark contrast to one of these helpless rats shown in black, where after a couple of minutes of movement, they'll quickly become immobile and kind of just float there, suggesting this showing this behavioral despair. So if we give one of these helpless rats ketamine, we give it injection 2 hours before the test. Now this helpless rat performs like the wild-type rat. It tries to escape the entire time. It moves around. You do not see this period of immobility anymore. And now if we can give the rat naltrexone before giving them ketamine, and just like that clinical study, this seems to completely block the antidepressive effects of ketamine. Once again, these helpless rats display this immobility. And so this data, I think, suggests, these are single rats, but the summary data suggests that yes, the helpless rats display this despair behavior, this immobility. Ketamine seems to be an antidepressant in this model, a rapid antidepressant. And naltrexone the opiate antagonist, does block this antidepressive effect. So we reproduced what they found in that clinical study. So we have this behavioral evidence of ketamine and being blocked by naltrexone. We wanted to look at a cellular correlate. So we decided to look at the lateral habenula. This is diagrammed here. The lateral habenula is this epithalamic nucleus known as a disappointment nucleus. For the purposes of this talk, it basically is thought to encode disappointment. If you're expecting a reward but you don't get that reward, activity in the habenula increases, it lights up. And this habenular activity, this increase in habenular activity inhibits release of dopamine from VTA neurons, so you never get that rewarding dopamine rush. So again, for the purpose of this talk, you can just think that more activity in the habenula leads to more disappointment, may lead to more depressive-like symptoms because you're not getting this rewarding dopamine release. And so therefore, any drug that reduced activity in the habenula may act as an antidepressant, because then you're going to feel that reward. You're going to have perhaps less depressive symptoms. Okay, so that's what we did. We wanted to start looking at what are the effects of ketamine in the lateral habenula. And the way we did this, this is a coronal section of a rat brain, and just some landmarks here. Up in the corners are the hippocampi. Here's the midline down here, the ventricle. And on either side here is a lateral habenula. And the reason they're green is because we've injected a virus that expresses a calcium indicator known as GCAMP. This calcium indicator, so when the neurons are firing, calcium is entering the neurons, and the calcium will bind to this protein, and the protein will fluoresce, will give off light. And we can measure the intensity of this light as a correlate for the neural activity. In this way, we can measure the activity of hundreds of neurons at once in the habenula. So this is what it looks like under the microscope. You can see the individual neurons, and they blink. And we can measure this blinking, the fluorescence intensity, as a correlate of neural activity and test to see what our drugs are doing. So how these experiments work is we're going to take a movie, and then we're going to wait 15 minutes either doing nothing, or we're going to put on a drug, and then we're going to take another movie and compare the intensity of the neurons in the movie before versus the movie after, and try to see if anything changed. So this is a graph of intensity over time of a single neuron, and the x-axis is time in seconds, and y-axis is fluorescence intensity. And you can see we take a baseline movie, and that's a black line, and there's some activity of the neuron. We're going to wait 15 minutes. We're not going to do anything and take the movie again, and that's the gray line. And then we're going to make a ratio of the intensity of the activity before and the activity after. And since this is kind of a control, we just want to make sure that our preparation is stable across these 15 minutes. Indeed it is. And so we take a ratio, and the ratio is about 1. So the intensity of the activity before and the intensity of activity after is the same. The ratio is 1. Now we can use some controls where we're basically putting in drugs that should decrease activity, either MBQX, which is an AMPA receptor antagonist. This is the primary excitatory neurotransmitter, so this will decrease activity. Or TTX, which is a voltage-gated sodium channel antagonist that should again reduce activity. And as a proof of concept, it seems to. So here with MBQX, there's some level of activity in black. After added MBQX, the activity decreases about 50%. You see the ratio goes down of post versus pre to about 50%. TTX reduces it even further, about 75%. So it looks like our system is working as we expect. We can measure activity and do pharmacology. So, okay. So what happens with ketamine? So again, as I said, we're looking at lateral habenular now. Agents that decrease habenular activity may be antidepressants, may have antidepressant properties. And so indeed, if we add ketamine, ketamine does cause a decrease in habenular activity by about 15%, suggesting that it is having an antidepressive effect at the cellular level in the lateral habenula. I think my keyboard stopped working. All right. If we add naltrexone being the opiate antagonist, now there's an increase in activity. We're not quite sure why is that, but importantly, naltrexone plus ketamine blocks this effect of ketamine. Now there is no longer this decrease in habenular activity. There is no longer this cellular antidepressive effect. So just like in the forced swim test, naltrexone, the opiate antagonist, seems to block the effects of ketamine. Damgo is an opiate agonist, so just activating opiate receptors does not have any effect on habenular activity. Now importantly, APV is another NMDA receptor antagonist. This has the similar effect as ketamine, lowers habenular activity a similar amount. Now most importantly, APV, the antagonist, plus ketamine while it reduces habenular activity, does not have any additional effect, suggesting there's an occlusive effect, that both APV and ketamine are working by inhibiting NMDA receptors. So in conclusion, ketamine acts as an antidepressant in helpless rodents, just like it acts as an antidepressant in humans, in depressed patients. These effects, both at the cellular and behavioral level, are blocked by naltrexone, the opiate antagonist. The effects of ketamine are mimicked by an NMDA receptor antagonist, but these effects are not mimicked by an opiate receptor agonist. So putting that all together, ketamine is not an opiate. Ketamine, we do not think that ketamine binds to opiate receptors. We think it is binding to NMDA receptors. However, opiate activity is necessary. You need an intact opiate system to produce these ketamine effects, but again, it's not sufficient. And so we think this means that the opiate system is permissive for the cellular and behavioral effects of ketamine somehow. We're not quite sure why. So future directions, very quickly. One, if anyone has any compounds, any novel antidepressants they think they have that they'd like to try in our system, we'd be happy to try them out. Two, the reason we think that this effect of naltrexone may occur is that there is actually a physical complex of the opiate receptors and the NMDA receptors in the habenula, and that's how they might be talking to one another. And we're looking into that. So I'd like to thank everyone that worked on this project, the Malino Lab, Robert Malino, Josh, our very talented research assistant, Salma, I don't know if she's here, but a very talented undergraduate, and also the folks that have helped in the psychiatry department, Neil especially, and I'd like to thank funding. So thank you.
Question.
Great system to study lateral habenular activity. I have two questions. One is about have you had a chance to check for calcium activity changes in other areas other than lateral habenular to see whether ketamine effect is specific? And the second is, are you planning to use a cell-specific driver lines instead of the pan-neuronal calcium GCAM6 viral approach, so that you could test for those projection neurons that from the lateral area inhibit the ventral tegmental area?
I think those are great, those are great ideas. We're trying to do both. We're looking at the prefrontal cortex as well as the VTA to try to get that whole circuit. And then, using viral-specific injections of the inputs and outputs. Oh, sorry. Yes, those are great ideas, and we are trying to do this. Yes.
Great talk, Matt. So in your model, it looked like the opiate agonism wasn't sufficient, but you said clinically that it's been known that opiate...
Yes, so that is true. I think the story is just a little complicated. Actually, maybe kappa antagonism that is causing the antidepressive effect. And so DAMGO is a mu-specific agonist. And I think we just have to look at some of the other receptor subtypes to kind of get to that question.
Okay, thanks.
But thank you. Yeah, it's an interesting question.
That was a really great talk, very understandable. My question is kind of similar to Davide's about the locus of the effect. And of course, there are receptors all over the brain, not just in lateral habenula.
Right.
So could you deliver the ketamine just to lateral habenula and test the behavioral effects?
So a group has done that, has done a cannula infusion into the lateral habenula and does observe an effect on the force swim test of either APV or ketamine directly infused into the habenula. We have not done that, though.
Neil, do we have time for a couple more questions? Okay.
Hi. Really nice talk. So my question is, if you can tell us more about the concentration and the dose that you use, because I can see that you use just one concentration in vitro and also in dose, in vivo, if you pick that one because you want to recapitulate was the dose that is prescribed in humans, or you actually do a dose response?
Of the dose of the ketamine?
Yeah.
So it's actually interesting. Rats metabolize ketamine a lot more quickly than humans do. So the rat dose is actually 10-fold higher than what you would give a human antidepressive dose. The dose we're using is 15 milligrams a kilogram injected IP. And it's actually, we can talk about this later, but it did take a while to kind of dial that dosing. I think there was sort of a U-shaped effect that too much, they're just too sedated and it's not effective, and too little doesn't work either. And also the route of delivery, there's this question of do you need this first pass metabolism? And actually the metabolites of ketamine are having the effect. So yeah, I think there's a lot to look at there, and a dose response and routes of administration. Thank you.
Okay, one last one.
Great talk, thank you. What do you think of the abuse liability effects of ketamine?
The abuse of ketamine, yeah, it's an interesting drug. So it is an abusable drug. In my experience of giving it to patients, most people don't seem to like it. The dissociative effect is particularly unpleasant. I don't know. We'll have to see if it's actually becomes a problem with abuse. I know that, so the intranasal spray that was just approved, it has to be given in a doctor's office under supervision. It's not like, oh, you can take it home with you, and this is going to be an antidepressant, a daily antidepressant you can take. So I don't think it's for that. I see ketamine really, its applications maybe in emergency settings, because it does reduce acute suicidality, and it does have this rapid effect. So it's something you could give an emergency department or an urgent care clinic, not something that you kind of take every day as an antidepressant because of perhaps the abuse concerns.
Okay, let's thank Dr. Kline one more time.
Thank you.
Okay, our next speaker is Joana Kay. She is a clinical psychology intern at the VA and UCSD. She is also a fifth-year doctoral student at Drexel University in Philadelphia. Her research focuses on exposure therapy for anxiety disorders, including factors to improve the dissemination and implementation of exposure therapy. Next year, she will continue at the VA and Sashi CSD as a clinical psychology resident in the Behavioral Health Interdisciplinary Program and Mood Clinics. Dr. Kay will present on what holds us back, the role of therapist factors in the cautious implementation of exposure therapy. Welcome.
Thank you for the introduction. So today what I want to talk about is what holds us back. So what are the factors associated with cautious implementation of exposure therapy? So we know that exposure therapy is very effective for anxiety disorders. However, it's dramatically underutilized by clinicians. So even when clinicians have been trained in exposure therapy, research shows they use this treatment in much lower rates than would ever be recommended by the existing research base. And what we also know is that even when therapists deliver exposure therapy, research shows they often deliver the treatment in an overly cautious way that really may impair treatment outcomes. So what do I mean by overly cautious? So here are some examples. Therapists might suggest deep breathing during exposure therapy, not allot very much time to exposure. They may inappropriately reassure patients during exposure. And so you might be wondering, they're doing exposure therapy, so really, do these behaviors matter? If we think about the goals of exposure therapy, two major goals are that patients can learn that they can tolerate anxiety without avoiding, and that the feared predictions they're making when they face their feared stimuli are actually fairly unlikely to come true or are more tolerable than they think. And these behaviors really may undermine some of that learning. So if a therapist allots a very small amount of time to exposure therapy, anxiety disorder patients really may not learn what they need to learn to really improve from their anxiety disorder. If a therapist suggests deep breathing during exposure, that really may continue to send that message that anxiety is intolerable, that it must be avoided in some way in order to move forward. And so these very simple behaviors really may undermine the mechanisms that we find exposure therapy to be so important for, and really may undermine critical learning here. So if we agree then that these behaviors matter, the next question becomes, how do we know who is using these behaviors and what we can do about it? So which therapist characteristics predict the cautious delivery of exposure therapy? So when you look into the literature, we see the following characteristics tied to cautious delivery of exposure. And we know that on average, therapists are more anxious when they're delivering exposure therapy than when they're delivering other evidence-based treatments. So that leads us to wonder, are they variables related to therapists' own anxiety that's interfering with effective exposure delivery, or might it be variables related to therapists' attitudes and beliefs towards exposure towards their patients? So within therapist anxiety, one related variable is experiential avoidance. This is the tendency to avoid or escape unwanted negative internal experiences, even when that might lead to harmful consequences. Another variable here is anxiety sensitivity. This is a fear of one's own anxiety reactions due to fears that they might have a variety of harmful consequences. Within attitudes and beliefs, we know that there's a lot of research tying negative beliefs about exposure to cautious delivery, including the idea that exposure is ultimately harmful, intolerable, or even unethical. Another related variable here is the perceived fragility of anxious individuals. This is the idea that if individuals with anxiety disorders are faced with intense distress, they'll decompensate or fall apart. It's based off of the spun glass theory of mind, that they'll break like spun glass. Another variable related to therapist anxiety is intolerance of uncertainty. This variable has been tied to therapists being less likely to use exposure therapy, so we chose to look at that variable here as well. So when we look at this literature, it's really in its infancy at this point. So consensus has not been reached about which therapist characteristics are most associated with the cautious delivery of exposure therapy. And a lot of this research is really based on self-report assessments of cautious delivery. So two of the major goals in this study were to determine, after therapists complete exposure therapy training, which are the characteristics that continue to be most associated with cautious delivery of exposure, And can we test that using both behavioral assessment of exposure delivery and self-report assessment? So moving to the procedures, we recruited psychology graduate students and mental health clinicians. This was part of a larger randomized controlled trial, who completed pre-workshop assessments, attended an exposure therapy training workshop, then completed post-workshop assessments of therapist characteristics, including a simulated exposure session that provided that outcome variable for us. So let's talk about what this simulated session looked like. So therapists attended this workshop. They were told to log on to Skype within 1 week of the workshop at a specified time. And at that point, they were told that an undergraduate had reported social anxiety symptoms and volunteered to participate in this study, and that this undergraduate was ready for their next session of exposure therapy. Their role was to deliver this session of exposure. Meanwhile, the Confederate actors were trained to exhibit a lot of signs of anxiety. So they fidgeted, they left long pauses, they asked whether time was up yet, they used a lot of safety behaviors. And our goal was to see, what do participants do? Do they respond by remaining silent or maybe encouraging the Confederates to continue? Or do they respond by reducing the intensity of the exposure in some way? Do they choose an easier exposure, stop the exposure altogether, suggest deep breathing? These were the behaviors we were coding in terms of cautious exposure delivery behaviors. We then gave some feedback to them and debriefed them about the deception. So our measures overall, then there were two for the quality of exposure delivery. So there's the behavioral assessment that I just described. And there's also a pen and paper measure called the exposure therapy case vignette. This is a hypothetical case vignette of a patient with OCD, where therapists are led to make a number of decisions about a session of exposure, including which exposure to do on a hierarchy, what to do when the patient exhibits some avoidance behaviors, things like that. And then in terms of therapist characteristics, you see the measures we used here below. A couple of points I'll make. So under experiential avoidance, we had a general measure of experiential avoidance and a measure of experiential avoidance specifically in the context of delivering exposure therapy. And at the bottom here, you'll also see the spun glass scale. So this is how we measure that variable of the perceived fragility of anxious individuals, that if faced with too much distress, these individuals will break or decompensate. So in terms of recruitment, we actually had no problem with. We had to shut down recruitment in only 4 days because so many people signed up to participate. So I think it shows how many therapists, despite some of this research, really do want this training. We ended up with a sample that was primarily female, white. We had 65% students and 70% endorsed a primary CBT orientation. So this was CBT broadly, including acceptance-based and behavioral orientations. So what did we find? So when we look at the behavioral assessment first, well, this is the p-values and the prediction of variance in exposure delivery by therapist characteristics using general linear models. And we find that the only variable associated with cautious delivery of exposure is this spun glass scale. So the beliefs in the fragility of anxious individuals. So none of the other variables tied in the literature to cautious delivery were associated with cautious delivery here on the behavioral assessment. But remember, we also had the hypothetical case vignette as our second measure here of this, of exposure delivery. And we see a pretty different picture. So we see that a number of variables were associated with cautious delivery on this hypothetical case vignette, including experiential avoidance during exposure, negative beliefs about exposure, and intolerance of uncertainty, as well as a trend with anxiety sensitivity. And again, we see this beliefs in fragility of anxious individuals scale. So this is the only measure that really cuts across both outcome measures and provides some multimodal agreement in terms of these different characteristics associated with cautious delivery of exposure. So to try to understand this a little bit more, I ran some correlations with the spun glass scale to try to determine whether there were actually a lot of relationships between some of these variables. And we find that there are. So there's a strong correlation between the spun glass scale and general experiential avoidance during exposure, and negative beliefs about exposure, suggesting that perhaps some of these measures are really picking up on a similar set of fears or potentially similar underlying construct in the context of exposure therapy. So one major takeaway here is really that the strongest evidence for the effect on exposure delivery in this study was this idea that anxious individuals are fragile and will break if confronted with intense distress. So therapists who strongly endorsed this set of beliefs were consistently across both forms of assessment, delivering more cautious exposure delivery. There's one other study to date that also mirrors some of this data using self-report primarily. And some of the problem with this finding is that it really flies in the face of a lot of data. So for one, we know that exposure therapy is highly effective and efficacious for individuals with very severe anxiety disorders. So that really goes against this idea that anxious individuals are indeed fragile and can't handle it. It also doesn't take into account the intense distress that individuals with anxiety disorders have often been facing for years and sometimes on a daily basis. So the likelihood that the day they come in to do exposure therapy to face their distress in order to feel better is the day that they decompensate and fall apart is quite low, and that's supported by data as well. We also have research that patients themselves find the idea of exposure therapy more tolerable than therapists do. So it speaks to this general issue that as therapists, we might over-fragilize patients, which is really concerning, especially in cases like this when it might ultimately lead the patients to not be receiving the most effective treatments, particularly delivered in the most effective way. And a lot of this research is mirrored when you look at rates of therapists choosing to use exposure in the first place. So it really highlights this idea that we need to maybe take a step back and think about some of the rationale we're using for the treatments that we choose. So remember, we also found some differential results on these outcome measures. So the case vignette had more variables related than the behavioral assessment. So one possible reason is that we did find significantly greater variability in scores on the case vignette than on the behavioral assessment. So it could be that the case vignette is picking up, is more sensitive to some of these relationships than the behavioral assessment. And so one hypothesis might be then that this idea, this belief that patients are fragile, might also be highly related to ultimately finding exposure to be harmful or intolerable, which then might lead patients to be, or therapists to be avoidant of facing some of those fears that arise for them, which together might contribute to cautious delivery of exposure. So what this might look like, so if a therapist is in a session with a patient and about to do an exposure, the therapist might start wondering, well, if he tries a harder exposure, will this patient fall apart? Might he drop out of treatment or have a panic attack? Can he even tolerate it? Is this treatment even ethical? So really what's going on is this therapist is viewing the patient as fragile, so endorsing these ideas that they'll decompensate, and that's resting on this bedrock assumption that the treatment might ultimately be harmful, intolerable, or unethical. So let's say that this therapist then chooses to reduce the intensity of the exposure in some way. Maybe he puts off the exposure to the next session or does a short or an easy exposure. Ultimately, what's likely happening is that the therapist is avoiding his own negative internal experiences about exposure. So perhaps the relationship between these variables is a sign that therapists aren't inherently just really anxious people, but rather that they're being anxious and avoidant of some very specific fears that arise when they think about doing exposure therapy. That patients are intolerable, or not that patients are intolerable, that exposure therapy is intolerable, that patients are fragile, and the treatment is ultimately unethical. And so that could speak to the general underlying concern that patients or therapists might view their patients as overly fragile. And that is something that we should consider targeting when we develop exposure therapy training workshops and in general help disseminate and implement exposure therapy in the most effective way. So thinking about some of the limitations and future directions here, we don't know the psychometric properties of the behavioral assessment. So one alternative explanation for these differential results might be that this behavioral assessment is not a great real-world test of actual exposure delivery in a clinic. It could be that this behavioral assessment also just has a lot more noise. So there was different confederates, there were a lot of variables, much more so than a pen and paper measure. We also do not know the psychometric properties of the domain-specific experiential avoidance measure used in this study. In terms of next steps, we need to further assess the effects of beliefs that anxious patients are fragile on real-world exposure delivery, as this was really one of the most critical findings in this study. We also need to further examine relationships between these therapist factors. So are these therapist factors actually picking up on a similar underlying construct related to the fragility of patients or not believing in exposure therapy, or are they all distinct and not at all causally related? The ultimate goal here is to reach consensus about which therapist factors are most predictive of cautious exposure delivery so that we can build training programs that effectively target those variables with the goal to ultimately do mediation analyses to determine if we can effectively target these therapist beliefs in the fragility of patients or whichever variables come out to be most critical. Does that then actually yield improved implementation of exposure therapy by therapists? So in conclusion, we know that exposure therapy is underutilized and delivered often in an overly cautious manner that really may impair treatment outcomes. And therapists who perceive their patients to be fragile really may not deliver exposure therapy effectively, and that's even if they make that decision to use exposure in the first place. And researchers should really consider targeting some of these therapist factors to improve therapist implementation of exposure therapy in this larger quest to ultimately help individuals with anxiety disorders receive the most effective treatments delivered in the most effective ways. So I want to thank you all for your attention, as well as give a thank you to the committee members for my dissertation, which was related to this project, my very large staff of people who helped me with this study, and of course the therapists who participated. Thank you all so much.
Are there any questions?
Given that your sample is mostly student therapists, do you see that as being a characteristic that may disappear once you have more seasoned therapists?
So it could be. So we were about 65% students, about 35% not. I didn't actually look at differences in exposure delivery between the student sample and the practicing therapists and did not find any differences. Interestingly, when you look at some of the baseline data, what we find is that students actually have lower negative beliefs about exposure and lower beliefs in the fragility of anxious individuals. And anecdotally, from running many of these assessments myself, that's what I found. It was actually the students who I think were really concerned about delivering it exactly correctly that did the exposures in a more kind of traditionally intense and less cautious way than it seemed for the many of the practicing therapists who I think sometimes use so many non-specific therapy behaviors like reflection and empathy and validation and all of that, that I would actually hypothesize the opposite, that it was practicing therapists and particularly older therapists who tended to use more cautious delivery behaviors. But it is definitely an empirical question as to whether if I were to use exclusively a sample of practicing therapists, might I have a different set of findings. Yeah, thank you.
Hi, thank you for a fascinating talk and very interesting subject. I was wondering, when you were looking at the therapist's perceptions, in addition to their anxieties about what may happen to the patient themselves, did you have any measure or sense of whether their own sense of self-efficacy, both in delivering the basics of the exposure therapy, but also in dealing with if this person does start to show signs of decompensation, a sense of self-efficacy, well, I know how to handle that. That is not a reason for me to avoid it.
Yeah, that's a great question. So after the fact, that was one of the measures that in thinking about this work, I wish I had included. Therapist self-efficacy is one of the variables in this therapist characteristics literature that people are looking at, the role of that variable. So I don't have a measure of that. I did have a kind of general measure of knowledge of exposure therapy that was not as significantly associated with the overall delivery of exposure, but it was a one question measure that I wouldn't put too much worth in. So I would definitely want in future research to look at therapist self-efficacy because I do think it could be quite important. Thank you.
Okay, we have one last short question.
I thought this was really cool, by the way, like really addressing something that's really important. It was interesting when you said that the older therapist had some maybe what you wouldn't expect, maybe even more fearful or whatever. I'm wondering if you talked about any like negative experiences with past exposure treatments. Because although I agree with you, I think exposure treatments also, increase the likelihood that you will have an adverse event in treatment with some. And I think of the problems sort of like with one trial learning events, like this, I had this one bad thing that happened to me many years ago, and then from then you're kind of biased moving forward. So I wonder if you got any data on if they've had any past negative experiences with, a session gone awry.
Yeah, that's a great question. It's definitely something I think to consider, especially future research that's using an existing group of practicing therapists using exposure therapy. My sample was primarily people who had very, very little experience of exposure or had heard exposure in their training and were interested in learning how to do it. So I would imagine that the vast majority hadn't done much exposure therapy prior to attending this training. So one of my thoughts regards to the older sample and their beliefs about exposure is simply training. Exposure therapy has become so much more researched at this point than, some of the people in my sample were in their 70s. So, I think that might be a piece of it. But I do think, if you think about, in the practicing community, who's delivering exposure therapy and then who is continuing to display these cautious delivery behaviors, it very well might be that they tried it once and they had a bad experience, or what they perceive to be a bad experience, and that's keeping them from being willing to give it a shot again. Yeah. Thank you.
Great. Let's give Joanna another hand. Okay, our next speaker is Dr. Giordano Guglielmo. He is an assistant professor in our department. His research focuses on interactions between neural circuits that modulate pain and addiction, and he has several high-impact research papers. He has more than a decade of experience in drug addiction research and has contributed to the development of unique experimental methodologies to investigate the neurobiological basis of drug dependence and identify novel effective pharmacotherapies for alcohol and cocaine and opioid addiction. Recently, he has identified and characterized a neuronal ensemble in the brain that controls alcohol drinking in dependent subjects. He will present on inactivation of a CRF-dependent amygdalofugal pathway that reverses addiction-like behaviors in alcohol-dependent rats. Welcome.
Thank you for the introduction, and thank you so much to the committee for selecting my abstract. This is going to be nice for us because we just moved to UCSD, so it's a good way to introduce some of the work we do in our lab. And particularly today I'm going to focus on neuronal ensemble that is CRF mediated that regulates alcohol dependence in an animal model, so alcohol abuse. So a lot of work has been done in the drug addiction field to identify the neurocircuitry that mediates the initial positive reinforcement after drug taking, but it has become increasingly clear that the transition to dependence involves the shift to a negative reinforcement phase that is characterized by the development of a negative withdrawal, a negative affective withdrawal state that in which the drug is taken only to self-medicate this negative affect state. And this phase is mediated by the brain regions that form the extended amygdala, and in particular the red circuit in here that it is characterized by the central amygdala, the BNST, and part of the nucleus accumbens. So today's talk is going to be focused on the central amygdala, and I'm going to talk about a neuronal ensemble that is activated in this specific brain region during alcohol dependence. So an important point in the drug addiction research, especially in preclinical model, is to have an animal model that mimics the human condition. And in case of alcohol abuse, it is important to have an animal model in which we can generate blood alcohol levels that are significant for alcohol addiction in humans, and that will be blood alcohol levels that are over the 200 mg%. And also, it is important to have an animal model that will generate the negative affect state during withdrawal that is characterized by anxiety and hyperalgesia. And also, it is important to develop an escalation of alcohol drinking. So this is what we do in our chronic intermittent ethanol exposure model, in which we train the animals to We train the animals to self-administer alcohol in the self-administration cages, where they are trained to lever press a lever and receive a reward of alcohol. And they will readily self-administer alcohol, but never to the point of being dependent and to reach super high blood alcohol level. So in order to do that, when they reach a stable baseline of intake, we put the animals in the vapor chambers. So in these chambers, animals breathe alcohol for 14 hours a day, and they go through 10 hours of withdrawal every day. So at the end of the 3 weeks exposure, they develop really high blood alcohol level that range between 200 and 250 mg%. And also they go through withdrawal every day. So they go through a withdrawal, acute withdrawal stage, where they show spontaneous somatic withdrawal signs, they show high anxiety, and they show hyperalgesia. So this picture, this is the animal 8 hours after the vapor goes off, and this is one of the most common somatic withdrawal signs called ventromedial limb retraction. And at the end of these 3 weeks, we start putting back the animals to self-administer alcohol in the self-administration cages during this acute withdrawal phase. And what we see is an escalation of alcohol intake, and after a few weeks, they drink twice as much as they were drinking before in the baseline phase when they were non-dependent. So as I said, these animals drink more, and we are interested on seeing what happens into the brain, like during this withdrawal phase, before they get access to alcohol. And as I said, we are going to focus in the central amygdala. Today is an area that regulates mood and stress, and there's a central role in the development of dependence. And we looked at the FOS activation in the central amygdala. CFOS is a measure of neuronal activity, and what we see is that animals that are in withdrawal from alcohol show high levels of FOS activation in the central amygdala during withdrawal. So a neuronal ensemble is recruited during withdrawal in these animals. And the first question we had was if this recruitment of this neuronal ensemble is causally related to excessive drinking, or only represents a consequence of excessive drinking. And in order to answer this question, we used a pharmacogenetic approach that is called the Dono2 inactivation method, in which we employ transgenic rats that are called CFOS LAX-Z transgenic rats. And in these animals, the LAX-Z transgene contains a FOS promoter that regulates the transcription of the LAX-Z coding sequence. And when there is a strong neuronal activity, such as alcohol withdrawal for us, the FOS promoter gets activated and leads to the production of LAX-Z mRNA and subsequently of beta-galactosidase, that is its protein. And at this point, we can inject a prodrug that is called Dono2 that is normally inactive in naive animals, but in this case, the Dono2 will be transformed by the beta-galactosidase in daunorubicin, and the daunorubicin will kill the neuron, leading to neuronal inactivation. So we use this method to see if by killing these neurons that are highly activated during withdrawal, we could lead to a behavioral effect. So we use the same model I explained you before with the self-administration training, vapor chambers, induction of dependence, escalation of drinking, and we implanted 2 cannulas in the central amygdala in this phase in order to be able to inject Dono2 after escalation of drinking. So what we found was that animals, as expected, escalated the intake during the ethanol exposure phase, and the injection of Dono2 blocked this escalation of intake. In fact, if you look, the alcohol consumed by the rats here went back to the levels that were pre-dependent, and this effect stayed for over 2 weeks. And also we found that the somatic sign of withdrawal were reduced. And when at the end of the experiment we sacrificed the rat and we looked at FOS expression in the brain, we found that the Dono2 group had a significantly lower level of CFOS positive cells compared to the control withdrawal rats. And the levels were comparable to a naive animal. So the next step was to characterize these neurons and see what kind of neurons they were. And the first shot we had was to do co-staining with CRF, because of CRF is really important in drug addiction, is well present in the central amygdala, and is the stress peptide prototype. So we found that there was no difference between naive rats and withdrawal rats in terms of CRF expression, but interestingly, 80% of our ensemble was composed by CRF cells. So it's important to mention that CRF is released in the central amygdala during withdrawal from most of the abused drugs. And if we inject a CRF antagonist in the central amygdala during withdrawal, we can decrease drug intake, especially on alcohol, nicotine, and cocaine. But all of this effect could be due to projections from the BNST LH into non-CRF neurons expressing CRF1 receptors. And until now, there is no causal evidence that the CA-CRF neurons are involved in this process. So, in order to answer this question, we generate a new tool that was the CRH Cray rat, in which the Cray is expressed under the CRH promoter, and this expression is only restricted to CRF cells. And with this rat, we can selectively target CRF neurons, and as we can see here in the video, we injected a Cray-dependent adeno-associated virus carrying a fluorescent protein, GFP. So in green, we will see the GFP, and in red is CRF. And we found that after injecting this protein and doing co-staining with CRF, 99% of the green cells were red cells, so 99% of CRF cells were also GFP. And also, this model allows us to use optogenetic to selectively inhibit these neurons. And this is an example with the electrophysiology, where animals were injected with this virus that is a Cray-dependent adeno-associated virus carrying an opsin that is sensitive to light, the allorodopsin, and also YFP. So we can target a fluorescent neuron with the electrophysiology, patch this neuron, and looking at this firing, applying the light, block the firing of the neuron, and as soon as the light is on, the neuron will not fire. And if we turn off the light, the neuron will keep firing again. And this doesn't happen in another neuron that was not fluorescent, so this is a non-CRF neuron. Application of the light doesn't lead to any significant effect on firing. So we use this methodology in vivo to target neurons in our withdrawal rats. And the first question we had was to see if inactivation of these cells has a role in alcohol drinking. So we injected the rats with the optogenetic virus. We placed 2 fibers into the central amygdala in order to shed light in the area. We trained the rats to self-administer alcohol, and inactivating the neurons in non-dependent rats didn't show any effect on drinking. So we moved to the next phase. We made the animal dependent, and we escalated their alcohol intake, and we did the experiment again. And in this case, inactivation of the CRF neurons in the central amygdala blocked escalation of intake. The effect was reversible, because if the light was off, the intake would go back up, and was like repeatable. We could repeat this several times. Also, the withdrawal signs were reduced. So we could mimic what we did with pharmacogenetic, also with optogenetic, and we could characterize which neurons we were targeting with pharmacogenetics. Next question was to see which projection from the central amygdala was required for drinking. And so we injected our virus in the central amygdala and looked at the terminal regions, and we found that these neurons from the central amygdala project to 4 major regions: the BNST, the substantia innominata, the lateral hypothalamus, and the parabrachial nucleus. So we injected the virus in the central amygdala, and we placed 1 fiber optic per terminal region. And we tried to inactivate the terminal region and see if we could recapitulate the effect that we saw in the central amygdala. So the first experiment was done in the BNST, and as we can see, by inhibiting the terminal region of the BNST, we could see the same effect that we saw in the amygdala in terms of self-administration and somatic sign. But this one was the only region, because when we inhibited the other 3 projections in the substantia innominata, lateral hypothalamus, and parabrachial nucleus, we couldn't see any effect. So we dig a little bit deeper into the mechanism using electrophysiology again, and the first question was to see if inhibition of these CA terminals was enough to inhibit a BNST cell. So we recorded from BNST cells, and in animals that were injected with the virus in the central amygdala, we turned on the light into the BNST, and we found that inhibition of the terminals was enough to reduce firing in a BNST cell. The last question was if this was a CRF-dependent mechanism. So we did the same experiment by recording from BNST cells in animals previously injected with the virus, and we found that application on the butt of a CRF antagonist could recapitulate the effect that we saw with the virus, with the laser illumination. And also, laser illumination on top of the CRF didn't lead to any other effect. So to summarize, I showed you today that alcohol withdrawal activates a CRF neuronal ensemble in the central amygdala, and pharmacogenetic and optogenetic inhibition of this ensemble can block withdrawal-induced drinking and somatic withdrawal signs. So these results identify a very specific pathway, that is the CRF-CA to BNST pathway, that could be targeted in the future for the treatment of excessive drinking in alcohol use disorder. So said that, I want to thank all my lab, and especially particularly Olivier George, my mentor, and Marcida, another assistant professor in the lab, all the postdocs, the techs, the graduate students, the undergrad, and all our collaborators, and the founding, and of course all of you for the attention.
question.
Thank you. This is really a great demonstration of the role of the CRH neurons in your alcohol addiction. I was wondering if there is any other neurotransmitter co-released with CRH in your region, terminal region. For instance, GABA maybe is co-released, and have you had a chance to inhibit GABA receptor specifically to see if their contribution is important as well?
So I can say that the CA is 95% GABAergic. So for sure is a GABA effect.
Yes.
And I didn't do that, but our neurons co-express dynorphin, for example.
Okay.
They co-express somatostatin. So we got lucky that we could find an 80% colocalization with CRF in our ensemble, and so it was easy to use the CRH CRERATs. But we thought to like try to inhibit using an shRNA CRE-dependent to see if it was a dynorphin effect or was a CRF effect. So it could be done. We haven't not done it yet.
Great, thanks so much.
Okay, let's thank Giordano one more time. And okay, this brings us to our plenary speaker for today. This is Dr. Jeremy Elman. Dr. Elman received his doctoral degree at UC Berkeley under Dr. Arthur Shimamura. His thesis research used fMRI to investigate contributions of the posterior parietal cortex to episodic memory. He remained at UC Berkeley for a postdoctoral position with Dr. William Jagast, using fMRI and PET imaging to study functional disruption and neural compensation in preclinical Alzheimer's disease. Currently, Dr. Elman is a postdoctoral fellow working on the Vietnam-era twin study of aging with Drs. William Kremen and Carol Franz. His work here has described genetic influences on imaging phenotypes and investigated measures of locus coeruleus dysfunction as an early indicator of Alzheimer's risk. He recently received a KL2 Career Development Award focused on characterizing the genetic and biological heterogeneity of Alzheimer's disease to improve our understanding of the early Alzheimer's continuum. He will present to us on the topic Cognition Predicts Progression to Amyloid Positivity in Amyloid Negative Individuals. Welcome.
So thank you, especially to the selection committee. I really appreciate the opportunity to present some of the work I've been doing recently. So I think the title kind of gives away the punchline of this talk, but really I'd like to focus on a more basic question, which is how can we improve early identification of Alzheimer's disease risk? So today I'll give some more general motivation. I want to go over a couple studies we've done in the Kremen lab to kind of get at this issue, then provide some more specific background for this current study, go over the methods and results, and then I want to talk about some implications I think that these results have for the field. So first, I think it's important to provide some motivation for why I think this question is so important. It's estimated that the number of Americans with Alzheimer's is really set to skyrocket in the coming years, so up to about 14 million people by the year 2050. But a question that often comes up is, if we have no effective treatment, what's the point of identifying people earlier? And in the Alzheimer's Association report from last year, there were some facts that I thought were really striking. So they estimate that among all the Americans alive today who will eventually develop Alzheimer's disease, if we were able to diagnose them earlier in that stage of mild cognitive impairment, or MCI, it could save $7 to $8 trillion. So translated to the individual level, that's about $64,000 per person. So we're talking about some really massive costs here. As many of you are probably aware, essentially all Alzheimer's clinical trials have ended in failure. Now, the reason that's often given to explain this is that we're initiating treatment too late. So there has been a move towards secondary prevention strategies, which means that we would initiate treatment after pathology appears, but before significant cognitive impairment. So we know that diagnosing in the MCI stage is important, but there is a complication here, which is that MCI is incredibly heterogeneous. So many people who are diagnosed with MCI never go on to develop frank Alzheimer's disease. So Eero Vuoksema, a former postdoc in the Kremen lab, really sought to improve our ability to predict which people in those MCI groups will actually go on to develop Alzheimer's. He looked at data in the Alzheimer's Disease Neuroimaging Initiative, or the ADNI, and so he looked at both cognitively normal, but also MCI subjects. And I should note that in the ADNI, they diagnose MCI based on the logical memory test. So Eero did something fairly simple. He just broke down this MCI group with an additional neuropsych test, in this case, the Ray Auditory Verbal Learning Test. And he found which MCI subjects showed normal performance versus somewhat impaired. So this figure on the bottom left shows that this AVLT impaired group actually had more abnormal amyloid and tau at baseline. The figure on the right shows that this AVLT impaired group also progressed to Alzheimer's disease at a much higher rate. So there is a question that remains, are these groups actually different? So it could be that both of these MCI groups come from the same risk population. The AVLT impaired are just further along. They're in a more advanced disease stage, so of course they're converting to Alzheimer's at a higher rate. But it's also possible that they actually reflect two different risk populations. The AVLT normal performing group could have quite a few false positives. So these are people that aren't necessarily on the AD continuum. We wouldn't actually expect them to develop Alzheimer's even if we followed them up for many years. So to address this, I computed Alzheimer's disease polygenic risk scores for all of these MCI subjects, and I found that the AVLT impaired group had much higher genetic risk for Alzheimer's. What this suggests is that these actually do reflect two separate risk populations, with those AVLT impaired subjects being more closely tied to the Alzheimer's continuum, and the AVLT normal performers possibly representing more false positives. So these are people who are just less likely to develop Alzheimer's in the future. So we are able to improve prediction of which MCI subjects convert to Alzheimer's, but ideally we're catching people very soon after they convert. So is there a way we can really improve that initial MCI diagnosis? Really the best way to do this is through repeat testing. Now, there is a complication in that repeat testing leads to practice effects. So people at follow-up will tend to do better simply because they've taken the test before. They may remember some test items, they may just be more comfortable with the testing situation. They could have picked up some strategies so they know that if someone starts reading them a list of words, they're probably going to have to remember them later. So looking in the Vietnam era twin study of aging, we tried to calculate practice effects. So I won't go into the exact methods, but people came in at an average age of 56 for baseline testing. They came back at an average age of 62. So this is a pretty long interval. It's 6 years. They're relatively young for an aging study. The figure on the left, on the y-axis, shows how we looked across multiple cognitive domains. And on the x-axis, we're giving the estimated practice effect. So how much of a boost in performance do we expect people to get simply because they've taken the test before? And the crosshatched bars, which are most relevant here, show that there are improvements of 0.2 to 0.3 standard deviations. And again, this is after 6 years. So that's a pretty big improvement. Now, the way we diagnose MCI it depends on whether people fall above or below some threshold of impairment. Now this boost could be sufficient to keep them above that threshold and could affect our numbers of MCI. So we took a look at this. So at baseline, about 11% of the sample had MCI. When they came back for follow-up, this actually went down to 9%. So that seems a little bit odd that there are less people with MCI. However, once we corrected these follow-up scores to account for this boost in practice, it went up to 15%. So we can also look at conversion rates, the percentage of people that are cognitively normal at baseline and progress to MCI at follow-up. This actually doubled from 4.5% to 9%. So a substantial number of people would have otherwise gone undetected, potentially until their next assessment. So moving to the current study, when you're talking about early identification of Alzheimer's disease, you have to talk about biomarkers. For anyone in the field, you've seen this figure in every talk. It's really been the predominant model of Alzheimer's disease based on the amyloid cascade hypothesis. And it really proposes that there's a fairly invariant sequence of events that occur. First, you get abnormal amyloid, then tau, neurodegeneration, and really only late in the disease do you see changes in cognition. And I do want to emphasize here that amyloid is thought to become abnormal years or decades before any changes in cognition. And I will say that there is quite a bit of evidence to support this model. Last year, the National Institute of Aging and Alzheimer's Association published their updated research framework. And this had a couple of components. So one aspect that sort of replaced this model of biomarker trajectories was the ATN classification system. So this simply categorizes people based on whether they show abnormal levels of amyloid, tau, or neurodegeneration. One nice thing about this is that it's agnostic to the temporal ordering. But they also propose a new staging of the Alzheimer's disease continuum. And this really does assume that standard ordering. So to even be considered on the AD continuum requires abnormal amyloid. And really in all cases, biomarkers are thought to become abnormal well before cognition. So again, as I said, there is quite a bit of evidence to support this ordering, but there's also quite a bit of data that seems to complicate matters. So the figure on the left shows some results from Brock and colleagues. They conducted a large autopsy study and found that actually abnormal tau appears quite early in life, in people's 20s or 30s, and essentially always appears before amyloid. So here we're already seeing that that ordering of Alzheimer's pathology may not be so clear. On the right, this is some data from the Mayo Group. They categorize their subjects based on this ATN system. On top, they're showing younger 65-year-olds, and they find that quite a few people show one single abnormal biomarker, and it's pretty evenly distributed across the three. When they looked at the older subjects on bottom, you now see a convergence, much more overlap, with quite a few people showing that classic AD profile of abnormal amyloid, tau, and neurodegeneration. So what this suggests to me is that actually people may start in different places, showing abnormal markers that could be different from each other, but eventually they're all taking different routes to the same endpoint. And as I mentioned, cognition is really thought to be affected only quite late in the disease. But again, there's quite a bit of data suggesting that this may not necessarily be the case. So it can change quite early in the disease. Judd and colleagues, in this figure on the left, they estimated when different measures become dynamic or start to show change. And they actually found that the AVLT may begin to change earlier than even other biomarkers. And changes occur in people that are amyloid negative. In predictive models, so using baseline measures to predict later cognitive decline, cognition often has effect sizes as large or larger than biomarkers. So it seems like cognition may actually be quite useful and sensitive in these early stages. I mentioned that trials have been moving towards secondary prevention, but we may need to go even earlier. We may need to initiate treatment before significant pathology appears. Yet we do still need a way to identify which people are most appropriate for intervention, so which are most likely to develop that pathology. Our assumption is that if the ordering of biomarker and cognitive trajectories is in fact variable, if it doesn't stick to that invariant sequence, each one has the potential to predict progression on the others. So other studies have looked at people with a range of baseline amyloid levels to predict which people will develop future cognitive decline. We're really doing the inverse. So we're seeing if we look at people with a range of baseline cognitive performance, can we predict which people will go on to develop abnormal amyloid? So I looked at 292 subjects in the ADNI. Importantly, everyone was amyloid negative at baseline. Then grouped them based on whether they showed progression to amyloid positivity at some point during follow-up. This could be determined by either CSF or amyloid PET. Then I looked at two baseline measures of cognition, the preclinical Alzheimer's cognitive composite and the ADNI memory factor score. I also controlled for some covariates relevant to cognition and amyloid. 41 of these 292 did end up converting to amyloid positivity. Otherwise, the groups are fairly similar. I want to point out that there were subjects who had mild cognitive impairment at baseline, but again, they were all amyloid negative at that time. So here's our primary analysis. If we look at baseline cognition, can we predict who will later convert to amyloid positivity? I did include a dichotomous variable indicating whether or not they had abnormal levels of tau. This is really the other hallmark pathology of Alzheimer's and could be associated with lower cognition. So in both cases, it turned out that baseline cognition predicts amyloid positivity. So lower baseline cognitive performance is associated with increased odds of progression to amyloid positivity. I do want to note that this dichotomous measure of tau abnormality was not significant here. But this leaves an open question. So these people did not surpass the threshold for abnormal amyloid, but that doesn't mean that there's no pathology present. So it could be that subthreshold pathology is contributing to this. So to address this, I included variables, continuous values of CSF, amyloid, and PET. And here what I found was that more abnormal amyloid predicted later progression to amyloid positivity. So perhaps not surprising, but again, remember, it's still subthreshold levels of amyloid. Interestingly, tau, when included as a continuous variable, was significantly predictive. But even after controlling for both of these biomarkers, both of our cognitive measures still significantly predicted progression. So it contains some independent and quite useful information. Okay, so what can we take from these results? I think the first is that subthreshold amyloid appears to be quite relevant. So often amyloid is looked at just in terms of negative or positive. The exact value isn't thought to be that important. It turns out it may be. And in fact, we may want to lower those thresholds if we're studying the early disease continuum to pick up some of that lower level pathology. I thought it was interesting that the dichotomous versus continuous values of tau could have led us to very different interpretations. So this could be due to just the increase in power you gain with a continuous variable. It could also speak to the relevance of subthreshold pathology. There are absolutely times when you need to use dichotomous values, say as inclusion criteria. But when possible, we think it would be very important to also look at these values continuously. Then I'd like to echo some previous findings that measures of amyloid accumulation over time can be quite valuable, especially in these early disease stages. Although someone might not have met the criteria for abnormal amyloid, if they're tending to accrue that pathology over time, we can be reasonably confident that eventually they will. And yet we've been able to identify them earlier. So I told you that cognition predicts amyloid positivity, but why would that be the case, especially when cognition arises from the brain? And I think there are two possibilities that are most likely. The first is that this lower cognition represents an actual decline. So in these subjects, disease processes may have already begun, and it's really the subthreshold pathology that's already there causing that decline. It's simply that cognitive measures are more sensitive than our current biomarkers. As detection thresholds lower, results like this may become less likely. However, it is also possible that the low cognition represents long-standing differences. These people may have always had lower performance, possibly due to less efficient processing, which would require more neural activity. And there is actually evidence that greater neural activity leads to increased amyloid release and deposition. So the lower cognition could either reflect that the disease processes have already begun, or it could flag individuals who are most likely to develop pathology in the future. And last, I'd like to touch on some implications for the NIA-AA framework. It's likely to be very influential in the field and guide research for many years. So there's been a move to define Alzheimer's as a biological construct. I think I may have less of a problem with this than some of the people in this room. But I think cognition is important to keep in mind. It really is the aspect of the disease we care most about. But it turns out it's also very important and valuable as an early identifier, potentially earlier than other biomarkers. When it comes to the staging system of Alzheimer's, again, I do think it accurately describes the way the typical patient progresses through the disease, but not everyone. So we found that some people who were amyloid negative at baseline had elevated tau or even MCI. Now, they would not be considered on the Alzheimer's continuum. When they then later develop amyloid, all of a sudden they're jumping onto that continuum at a much more advanced stage. So I think we need to maintain some flexibility and allow for possibilities such as this, that people may not progress from stage 1 onwards in a linear fashion. And then last, I'd like to talk about whether we use amyloid positivity as inclusion criteria for our studies. There has been a move towards requiring this in some cases. Certainly think it makes sense. In something like a clinical trial, especially when targeting amyloid, you need to be sure that there's amyloid in the brain if you're going to try to target it. But if our purpose is really to understand the early AD continuum, I think it might make more sense to have more inclusive criteria so that we can detect, identify, and really explain these factors that can lead to variable progression. So before I end, I'd like to thank the many people I've been fortunate enough to work with here at UCSD. In particular, I've received daily mentoring from Bill Kremen, Carol Franz, Matt Panazon, and Chris Fenneman-Oatstein. Again, I'd like to thank the selection committee, and I'd like to thank all of you for your time and attention. I can take any questions.
Questions?
This is a great talk, Jeremy. Thank you for focusing on behavior.
Thank you.
Have you looked at any other behavioral measures in ADNI? I see that you kind of expanded within a memory domain. What about the non-memory domains to try to capture those MCI subtypes that are non-amnestic at first presentation?
Yeah, so I think that's a very good question. Part of the reason for that is that in ADNI, they really select for amnestic MCI, so they're essentially really focusing on people with memory impairments. In the VETSA, where we aren't selecting for that, we do have non-amnestic MCI, and I think it would be very important. Another postdoc in the lab, Dan Gustafson, has found that executive function can be very predictive of later conversion. So that is absolutely something I would like to look at.
Jeremy, thanks for a great talk. You mentioned therapies that target amyloid. The world is appropriately disappointed with the failure of the Biogen trial. Could you comment on further on amyloid targeting therapies and how it fits in with MCI, early identification, integration with cognitive measures.
Yeah, and that's, kind of the difficulty here is with, I think Mark Bondi, I'm not sure if he's here, but I remember in a talk he mentioned this, that we've been getting all these failures of amyloid trials, but we're almost doubling down on amyloid as the key component. So again, to be on the AD continuum requires amyloid. And again, that may be the case for some people, but Alzheimer's is very heterogeneous, very complex. Brock and some others have suggested that tau may be the initiating event, so it's still not clear if amyloid is actually responsible for setting off this whole cascade or if it's a byproduct. I think it's quite likely successful treatment will require targeting amyloid, tau, neuroinflammation. Really one of the most effective treatments at preventing MCI has been physical activity or lowering blood pressure. So the Sprint Mind study found that lowering blood pressure reduced rates of MCI. So yeah, I think we'll need to move towards an approach of using multiple drugs, almost a cocktail. So far, the approach of targeting amyloid does not seem to have worked, and it's hard to see how that's going to change.
Thanks, because I was in that cohort in your first slide showing the rising rates of Alzheimer's by 2050. So.
Okay, great. Well, let's thank Jeremy and.
So I'd like to thank our speakers and questioners and Jyoti Mishra for doing a great job running this session, literally running around the room with the session. So thank you, Jyoti. So I think you've just seen an incredible breadth and depth of the research topics that we cover in our department, different stages of training and early career paths, rigorous approaches to many different aspects of science across different disciplines. And just wait until what you're going to see in the posters as we adjourn to the poster session in just a minute. I'd just like to remind people that if you're in posters 1 to 52 or mentoring that group, you mentor first and eat second. And if you're in the higher numbers, you eat first and mentor second. And so we're not going to check your number when you go get the food, but that'd be great. I also want to do my best channeling Dr. Grant by saying, everyone please clean up your trash here. Don't leave any trash with you. Make sure to take your programs with you if you can to the other room. And thank you again for a wonderful morning session. Oh yes, and remember to vote for the Best Poster Award. I will want to say that the trainees this year asked us to do something different, two things different. One is to have the Best Poster Award voting process, and you'll see that as you go in. But secondly, the mentors were selected to mentor particular posters because they don't know anything about that aspect of the field. So this isn't the situation where the mentors are going to be really smart and tell you all the new stuff. This is going to be where they're going to be, sitting there and trying to do their best to understand what you're telling them. So have pity on your mentors. They're doing the best they can. Thanks, everybody.
So, Neil, can I do one last comment as the exiting chair? First of all, to congratulate the speakers and everybody for just a fabulous session, and also to congratulate the mentors for helping these trainees do such great work, A. And B, I really did get a whipping from Mia Savoya when this room was left trashed by a previous psychiatry thing. Now, maybe you would enjoy seeing me be whipped. That's a whole other thing. Please, I want to avoid that.